Hello, everyone, and thank you for joining us again today for our workshop on the cell biological basis of epithelial physiology. Yesterday, we had a terrific set of panelists and discussions that covered topics ranging from understanding the barrier function of epithelia, their integration across scales, and the role of epithelial in health, disease, and in tissue self-organization. Today promises to be equally exciting. Like yesterday, it will consist of 4 sessions, with each session consisting of a few 10-minute talks followed by a 30-minute discussion. Like you heard yesterday, these discussions will not just be Q&A. Instead, the discussion leaders will engage in a conversation amongst themselves, the other panelists, and with you. Please use the Q&A box to submit comments or questions that you'd like us to address in the discussion. We hope to invite many of you on screen to join the discussion and the conversation that we're having. So without further ado, I want to now turn the floor over to my co-organizer, Michael Caplan, who's going to be introducing our first session. Michael.
Thanks, Jennifer, and welcome again, everybody, for what promises to be a really exciting day. The first session is entitled How Cells Integrate Chemical and Mechanical Signals in Space and Time to Drive Tissue Morphogenesis, Regeneration, and Physiological Functions. And at this point, I'll turn the leadership of the session over to our discussion leaders, Kathy Green from Northwestern and Prisca Liberali from FMI Basel.
Thank you very much, Michael, and welcome everyone. Today it's my pleasure to kick off this session. We'll first be hearing from Jen Zallen, who is going to be highlighting some important questions in epithelial morphogenesis that I think can be applied to any system, but she will be talking about it through the lens of a model organism. And then we'll be moving into the mammalian liver, and we'll be hearing about some exciting new findings from Marino Zerial's group, who's answering a fundamental question in membrane polarity and the formation of polarized membrane structures in that context. And then we'll move on to Kristin Knouse and Sarkar, who are going to continue the theme of mammalian liver, and we'll be hearing about their work using it as a powerful model of regeneration and also a way to look at the impact of nutrition on epithelial tissue homeostasis. And now I'm going to hand it over to Prisca, and she's going to be posing some questions for you guys to think about during the session. Prisca?
Yes, thank you, Kathy. And then, yes, during the talks, we would really like you to think of some aspects that we are considering and that we have been discussing in preparing this session. And, you know, how to challenge the status quo and think how to push the boundaries in some of our experimental settings. The first one would be to try to visualize and perturb the system at different biological scale, really trying to cross biological scale from molecular to cellular to all this, the way to tissue and organ scale properties, and to include gene expression, signaling, metabolic identities, and especially also mechanics. The other aspect that we would like you to think about that would come back also during the discussion at the end is also about tool development to enable large-scale analysis beyond gene expression and which type of tool that could bring us together as a community. So without further waiting, I will introduce the first speaker of the session. So the first speaker is Jennifer Zallen. She's an HHMI investigator from the Sloan-Kettering, and the title of her talk is Signals, Forces, and Cells, Decoding Tissue Morphogenesis. All yours, Jennifer.
Great. Thanks very much for the opportunity to participate in this exciting meeting. So I will share my slides. Can everyone see them okay? So, okay, great. Today I'm going to talk about the problem that we work on, and then I'll tell you about some challenges that we face in our research, and I'll propose 3 possible solutions to address these challenges. And if Janelia were to take on any one of these, this would be a huge advance for the field. Okay, so the problem we work on is how do you build a three-dimensional tissue structure in a multicellular organism? And this problem is important because the function of every organ in the body depends on its structure. And this problem is also hard because organizing hundreds or thousands of cells in time and space is a complex and physical process. So one way to think about this problem is to imagine a crowd of people at a busy train station. Each person has somewhere they need to go, but how they get there can be quite complicated. So to get where you're going, you not only need to know where you are on a map, but you also need to take into account the movements of the people around you. Similarly, cells in the body often find themselves in crowded environments where each cell needs to generate force in order to move, and it's also exposed to forces generated by the cells around it. So this slide here, this movie shows a movie of axis elongation in the Drosophila embryo, which we study in my lab. And during this process, organized cell rearrangements cause the body axis to double in length from head to tail, which is left to right in this image. And this process is driven by spatially regulated cell rearrangement. So individual cells within this tissue are moving up and down in a polarized and coordinated fashion. Now, these movements are generated by forces, and these forces are spatially localized in cells by the localized activity of the non-muscle myosin 2 actin-based motor protein shown in green and the par3 polarity protein shown in red. So in this movie, time is indicated in minutes and seconds, and you can see that as the cells get ready to intercalate, par3 in red becomes restricted to the horizontal edges of cells, and myosin is recruited to the vertical edges of cells, where it generates contractile forces that drive cell rearrangement and axis elongation. Now, we'd like to understand how this process works, and this process is generated by force. And actually, in our lab, we've discovered several molecules that control where and when these forces are generated in the cell in the embryo, including proteins that are at the cell surface in the toll and neurin receptor families. But today I'm going to talk about how cells respond to force. And there's evidence emerging that forces are not only fundamental drivers of cell shape and tissue structure, but forces can also activate biochemical signaling pathways in cells, just like chemical signals do. So for example, so when you think about how cells respond to forces in vivo, it's really helpful to think about insights that have been obtained from studies of how proteins respond to forces in vitro. And work from many labs have shown that applying mechanical forces to proteins can stretch these proteins and change their conformation, which can lead to changes in the protein's binding partners or its phosphorylation. And these are expected to have consequences for protein localization and activity. For example, work by Yas Sawada, Masako Tamada, and Mike Sheets has shown that stretching a protein in vitro can increase its tyrosine phosphorylation. However, while we know a lot about how proteins respond to forces in vitro, we know much less about how cells respond to the physiological forces that they see in the embryo. And this is a difficult problem to study because while we have excellent tools to study genes, RNAs, and proteins in the embryo and animal, we have many fewer tools to study forces in vivo. So we would need better biophysical tools to both measure and manipulate mechanical forces in the animal. So forces are, they can act extremely fast, they can be local or global, and they can be transient. So in order to visualize how cells respond to forces, we also need better molecular tools to watch dynamic processes that are happening in a very temporally specific way. And finally, it would be great to have better imaging tools to try to address these questions in mammals. So I'll tell you a little bit about some of the progress we've been able to make using current tools. So when you think about where mechanical forces are active in the cell, they're not uniformly distributed in the tissue. So in particular, in epithelial tissues, forces are predicted to be highest at multicellular vertices where 3 or 4 cells meet, shown here in green, and lower at bicellular junctions where 2 cells meet. So if cells have a way to respond to mechanical forces, then that response is predicted to be particularly active at these multicellular junctions. So to understand how cells respond to forces in vivo, Huapeng Yu, a postdoc in the lab, developed a method to visualize tyrosine kinase signaling live in the embryo by injecting embryos with an antibody to a fluorescent conjugated antibody to phosphotyrosine. And in this movie, you can see that the phosphotyrosine signal is highest at these multicellular junctions that are predicted to be under the highest tension within the tissue. And this process is, this localization of phosphotyrosine is tension dependent. So when he inhibits myosin activity by injecting embryos with an inhibitor of its upstream regulator rho kinase, you can see that in contrast to wild type embryos, which are referred to as high tension here, where phosphotyrosine accumulates at tricellular junctions, phosphotyrosine now spreads out over all junctions in the presence of low tension. And using by screening a series of a publicly available collection of GFP tag proteins, HP was able to discover the molecular basis of this tension-sensitive tyrosine kinase signaling pathway, and he showed that the Abl tyrosine kinase phosphorylates the actin-binding protein canoe, and this it recruits it to tricellular junctions and is required to stabilize cell adhesion under tension. Okay, so we're able to discover this mechanotransduction pathway because we can inject antibodies into the Drosophila embryo. But in order to study other organisms and other stages of development that are less accessible, it would be great to have better biophysical tools to not only decrease tension, but also to increase it and apply it in a localized and controlled fashion. And because we don't really know the full range of signals that respond to mechanical forces, it would be great to have more tools to visualize signaling live in the cell to try to understand how these cells are responding to forces. For example, it would be fantastic to have genetically encoded tools to visualize changes such as nanobodies, to visualize changes in protein conformation, protein phosphorylation, and protein localization in response to forces. And these tools could also be used to study other biochemical signaling pathways and could be applied to many organisms. And the third thing I'd like to talk about today is how do we apply some of the concepts that we've learned in model systems to study mammalian development. So this slide shows the process of neural tube closure in the mouse, which is an important problem for human health. So the neural tube starts off as an epithelial sheet that folds to form a tube along the length of the animal, and that will go on to form the brain and the spinal cord of the animal. And this process is highly dynamic and force intensive, as you can see in this movie by Eric Brooks in the lab. So we would love to understand, have a similar understanding of how do biochemical and biophysical signals work together to build this structure. And now is really a great time to address this problem, because we can take advantage of a large amount of information that's being generated about transcription in the mouse embryo, including single cell RNA sequencing studies showing where and when every gene is expressed in the embryo, as well as microscopy advances at Janelia that now make it possible to track cell behavior at single cell resolution in the developing mouse embryo. So what we'd like to do is to fill in this gap between transcription and tissue organization and function. And to do this, we need to define all of the molecular events that build the animal. And it would be great to have tools to visualize proteins and signals live in living cells. So for example, if Janelia were to take on the effort of tagging every gene at the endogenous locus in mouse embryonic stem cells, these cells could be a great resource that could be used to study many different kinds of cell types in vitro through directed differentiation. They could be used to study organoids, and most importantly, they could be used to visualize protein localization and signaling events as they happen in real time in living animals. So I just want to summarize. These are the three tools that I talked about today that would help move the field forward, including biophysical tools to measure and manipulate forces in the animal, genetically encoded tools to visualize all the changes that are happening in cells as they respond, and tools to study protein localization in mammals, so we can take these questions to a mammalian system. And I just want to acknowledge the people in the lab who made important contributions to this work, as well as thank HHMI and the NIH for funding.
Thank you very much, Jen. That was fantastic. I'd like to now introduce the next speaker, Marino Zerial, who is from the Max-Planck Institute of Molecular and Cell Biology and Genetics. And the title of his talk is Anisotropic Expansion of hepatocyte lumina, and liver tissue morphogenesis. Marino.
Yeah, thank you, Kathy, and thank Prisca and everybody for organizing this meeting. It's fantastic. So I'd like to come back to the question that was posed at the beginning, actually introduced by Kathy and Prisca, the problem of self-organization propagated along scales, because that's really this is our problem, understanding how, from molecules you get to subcellular, cellular structure, how cells from tissue, organ are organized. This remains a formidable challenge for all of us. And I'd like to walk you through really some of the work that we have been doing to address this problem in the context of the liver, because I think you will see more and more about the liver here, also in this session, because it's actually a very interesting organ and it's not so well studied as others from a cell biological point of view, I'd say. So this cartoon shows you a liver lobule, which is in a way really the unit that forms the liver. And basically the liver, the lobule, is a two-pipe system if you want to really having an hydraulic type of definition. So you have blood that comes really from the portal vein and the hepatic artery, and then goes into a capillary system, which is called the sinusoid, and exit via the central vein. Let me just, so via the central vein. And then you have the hepatocyte, they secrete bile, and the bile, of course, will be going to bile ducts, and the bile from the bile duct will go into the gastrointestinal tract. So it's an incredibly interesting system from many points of view, the many cell types that compose it, the particular architecture of this cell type. One thing we like to contrast to you is the hepatocyte polarity, so the major cell type of this liver lobule, and the classical simple epithelial polarity. That's what, for example, of the bile duct cells. So these bile duct cells, the cells of the form, the epithelial cell from the bile duct, have an apical surface that is facing the lumen of the bile duct. That's where the bile generated for the hepatocyte flows out of the lobule. So this is a typical epithelial organization and typical apical-basal polarity. The hepatocytes are peculiar. And this is an interesting point. So the hepatocyte actually have an apical surface that faces the apical surface of another hepatocyte. So two hepatocytes together, juxtaposed, form this bicanaliculi in 3D, and these are sandwiched between sinusoidal endothelial cells. So in 3 dimensions, this hepatocyte look like this. This is, it has a potato shape, so to speak, but you have this green surface, which is the apical surface, which is not one face of a cell, but like a ramified belt around the cells. And similar, you have a similar complex structure of the sinusoid that face this hepatocyte. Therefore, these cells is not like the classical simple epithelial cells. They have a polarity which is in a way multipolar. And altogether, that's the reason why the tissue assumes this morphology in 3D, a very 3-dimensional network of bicanalicular sinusoids. So now you can measure this actually in vivo by intravital microscopy. You see here bile flowing green, flowing from the portal to the central vein in this direction, and blood, we wish we had faster imaging, you will see blood following in the opposite direction. So just to illustrate this pipe system of blood and bile in the liver. Of course, the question, how they form. And I insisted on particularly the problem of polarity, and Mike Caplan knows very well really how much of this work has been done really in many systems to understand the mechanism of apical basal polarity. Unlike the classical simple polarity, this hepatocyte, as I said, have a more complex polarity, have a belt of apical surface, and that can be defined by nematic tensors. So different axes, basically you need at least 2 axes to define this polarity. So that's why this belt can be defined in essence by a multipolar system. If you now look at the tissue, you realize by some averaging that there is some logic how the tissue is put together. So the hepatocytes are actually oriented. The apical surface is oriented. And this orientation you can see here between central and portal vein is like a liquid crystal. So you have you have local noise, but you have overall distance. There is overall order along distance. And this polarity also follows also the orientation of the sinusoidal network where blood is going through. In other words, really, these hepatocytes in sinusoidal cells are configured together so that they can form pipes which are pretty much oriented along the two veins axis. So therefore, it's interesting to think that there is a mesoscale level of organization for the tissue. We can talk of mesoscale because the lobule is part of the organ, and so is a tissue structure within the organ. So there is a liquid crystal organization at the lobule level, which is due to local cell-cell interactions, as well as weak long-range cues. I'm saying weak because this is not the crystal structure, it's a liquid crystal with some freedom in the short range. And therefore, this long-range cue must be mechanical and or chemical, and probably both in the case of the liver. There are also multicellular structure because you have hepatocyte, sinusoidal endothelial cells, and stellate cells that form again some mesoscale units within the liver lobule. And of course, all together, then they form the lobule. So we think that there are mechanical forces, tension, pressure, fluid sharing. I told you there is, in fact, these are pipes that transport blood and bile. That these forces that propagate through the tissue, and they make, therefore, this is one of the team yesterday, they make tubes instead of spherical cysts as you have them in vitro. So in vitro, for example, of course, you have the sinusoid, but in vitro, bile duct cells will form cysts, will not form really pipes, tubes. But the bile canaliculi can. And let me show you, in fact, if you culture hepatoblasts, which are the fetal precursor of the hepatocyte, what you find is they form, this is the apical surface of the hepatoblast differentiated into hepatocyte in vitro. And they form this bicanalicula in vitro. You don't need tissue stretching, you don't need, of course, they are not well oriented, they are having, chaotic, but they nevertheless form this canalicula, this lumina. And it turns out, you've seen also from the before, you see these stripes labeled by actin here. We were very curious about that. And we looked also at the ultrastructural level. It turns out that there are projections between these cells. This is the apical surface of cell 1 facing the apical surface of cell 2. And you find these connections between them. We think that they're very interesting. They are not actually philopodia-like connection. 3D reconstruction of serial sections show you that these are actually like septa between the cells. They are junctions between the cells. And now I use this scheme just to illustrate how they look like. When you look at this reconstruction, this is idealized, right? You find that this is really how this apical surface between these two juxtaposed cells look like. You have these projections, this apical surface, they go into the lumen, and the two the coming from the green or the blue cells are sealed by tight junction, which are the T-shaped. So we think that these are actually similar to the bulkheads of boats and planes. These are really mechanical elements that keep the two cells juxtaposed so that as the grumen grows, it will not form a cyst, but it will stay together to form a bicanalicular. So these, we think, are very important elements to specify how the hepatocyte will look like when they polarize. So of course, that means at the cell level, at the cellular level, these are cell autonomous mechanisms because an hepatocyte is an hepatocyte and not the bile duct cells, but there are also local cell-cell interactions. So the bulkheads, as we call them, are mechanical elements that would therefore introduce anisotropy in apical surface tension. Basically, you will have tubules which are formed in vitro without tissue cues, which are the mechanics of tissue at the tissue level, in contrast to the epithelial tubes, which can't form so easily in vitro. In fact, that's why you have the formation of cysts. By doing a screen, we could actually convert this hepatocyte polarity into, in essence, bile duct cell polarity. We were lucky to find that a number of genes, but the most impressive one is the knockdown of one gene, or one of our gene, which is RAP35. And I'll show you the RAP35 converts this bicanalicular hepatocyte polarity into a simple polarity. Here, you start forming cysts in vitro instead of the classical. You see this in this cartoon, that these cells now form this dome that really come from the bottom of the plate, showing that they are changing polarity. And in fact, with Rab35 knockdown, what happens is that we lose the connections, these bulkheads, and therefore the cells are no longer able to form this tubular lumen, but they expand, and now they share, multiple cells share the same lumen like you have in simple epithelia. And you can see therefore that at the molecular level now, how to explain this, we know that there are various molecular pathways. You have Rab35, as I told you. In fact, there are vesicles, very likely recycling endosome at the base of this projection that you find between the two cells, these bulkheads. There is actin, as I show you here, also by super resolution microscopy. You have junction protein, which are T-shaped, so it raises the question whether they have special junctions, protein expressed in these cells. And of course, we must have mechanosensing mechanisms, which have to be somehow feedback into the molecule. I'm just showing you this because by using Rab35, we could re-engineer the tissue now. That means really not. Yes?
At the time, try to.
Okay,
we're already 1 minute late.
Sorry. Yeah, sorry, I'm almost done. So we can re-engineer the tissue so that in vivo also it can form bile duct-like structures. The hepatocytes now are not just screwed up in polarity, they are changing polarity and they form bile duct structures. So then going back really to the problem of self-organization propagated over scales, I like to finish with the fact that we have to study organelles, subcellular level, cellular and cell-cell interaction, and of course, large scales like this. And at the end, I go back actually to what Thomas was saying yesterday. We have the problem of thinking about how this interplay between chemistry with the molecules, mechanics with the forces, and architecture, size and shape are interplayed all the time and throughout all these scales. Really, this is a feedback system between these three elements and throughout the scales. And of course, that is interesting because it does determine also cell fate decisions. Thank you.
Thank you very much, Marino. Thank you. Really, this was great. And then it's my pleasure to introduce the next speaker. The next speaker is Kristin Knouse. And Kristin is from the Whitehead Institute, and she will give a talk speaking about delivering insight into organ homeostasis and regeneration through in vivo genome-wide screen. I would just remind you to please, you can post your question on the Q&A button, and we will address them at the end.
Thank you, Prisca, for that introduction, and thank you to the organizers for this really great opportunity for the field to come together and look forward. I'm excited to tell you today about our work to bring genome-wide CRISPR screening into the mouse liver to allow us to do high-throughput genetic dissection of organismal physiology and disease. And so the fundamental question that my lab is interested in is what underlies the regenerative ability of select organs in our body, and can we somehow confer this to different tissues in the setting of disease? And so if we think broadly about the regenerative ability of the organs in our body, we typically classify them into two extremes. There are those tissues that can continuously renew owing to the presence of stem cells that generate new specialized cell types. And there are those that cannot renew because they lack stem cells and are comprised entirely of terminally differentiated cells. And a problem with these type of tissues is that if these terminally differentiated cells are ever lost, they cannot be replaced. And indeed, this principle underlies the lifelong morbidity associated with numerous diseases, ranging from heart attack to stroke, neurodegeneration, and diabetes. And a longstanding goal of regenerative medicine has been to be able to unlock proliferative capacity in these cells. But thus far, we've been unsuccessful reinforcing this notion that proliferation and differentiation are mutually exclusive. And that's where the liver really emerges as a remarkable and I think highly informative exception. Because while the liver has very low turnover at baseline, it has the ability to completely regenerate itself after injury. And importantly, this regeneration is driven not by a stem cell population, but rather by the proliferation of the differentiated hepatocytes responsible for this organ's function. And so these hepatocytes are considered to be quiescent rather than terminally differentiated. And I think this tells us some very important principles about organ biology relevant to efforts in regenerative medicine. And that is that cell cycle rest is not an obligate consequence of differentiation. and therefore organ regeneration is in principle possible without a stem cell population. And so we have within the liver the instructions for how to build a regenerative system. But thus far, we still do not have a molecular answer to the question of why it is that the hepatocytes can proliferate and the liver regenerate, while other differentiated cell types cannot. And so if you think about how we might go about asking and answering these questions, we could certainly take hepatocytes out of the organism and bring them into culture, where we would benefit from strong experimental tractability. Through transcriptomics and proteomics, we can perform high throughput observational experiments. And through chemical and genetic screens, we can make causal inferences, also in a high throughput fashion. But in bringing these cells out of the organism, we necessarily strip them from the many factors that naturally regulate their biology in their native context, including interactions with other cells in the matrix and the role of circulating factors. And so I knew for studying a process like regeneration, it was absolutely essential that we study it in the organismal context. But I also know that for studying this and so many other organismal processes in mammalian systems, we necessarily have had to sacrifice experimental tractability. Because while we have long been able to perform high throughput observational experiments through transcriptomics and proteomics, when it comes to inferring causal relationships, we've necessarily been restricted to analyzing the functions of individual genes at a time, typically through knockout mice. And so this has classically dramatically restricted our ability to really dive into understanding organismal physiology in a comprehensive and unbiased manner. And so I thought to myself, well, what if we could bring high throughput functional genomics into the organism and thereby bring the experimental tractability once restricted to cell culture systems into these mammalian models, where we could benefit, therefore, from high throughput perturbational experiments and the physiologic relevance. And so genome-wide screening using CRISPR-Cas9 has really emerged as a powerful tool for dissecting cellular phenotypes. The original approach involves lentiviral delivery of a guide RNA library to cells in culture, and typically this means delivering tens of thousands of guide RNAs to tens of millions of cells in culture to allow for the coverage required to detect changes in the abundance of guide RNAs over time. The cells are then expanded for many population doublings to allow for protein depletion and phenotypic selection, and then the guide RNAs are sequenced at the end of the screen to identify changes in their abundance and therefore hits in the screen. Now, in terms of bringing this into the organism, we've long suffered from the inability to deliver lentivirus to solid organs at an efficiency compatible with genome scale CRISPR screening. And so we've had to rely on other approaches that have necessarily restricted the scale at which we can do these types of experiments. And so thus far, CRISPR screening in tissues has really been limited to small scale positive selection screens on the order of tens or hundreds of genes. And so I wanted to overcome this delivery barrier and allow us to perform comprehensive genetic screening in a solid organ. And so my first goal was to try to bring lentivirus into the liver at sufficiently high efficiency. And so I spent several months in the mouse room just injecting mice of various ages in various ways. And what I found is that if I injected lentivirus into neonatal mice intravenously, I could actually transduce hepatocytes at high efficiency. And so we generated a very simple lentiviral construct in which we can express both a guide RNA and a fluorescent reporter. And so to get a sense of the efficiency of guide RNA delivery and the multiplicity of infection, I combined two different lentiviral vectors, one expressing an M-cherry reporter and one expressing an M-TRK2 reporter in equal amounts. And I concentrated this virus about 1,000-fold and then injected between 2 and 50 million transduction units intravenously into postnatal day 1 mice. What you can see is that in this dose range, we can achieve a dose-dependent transduction in increasing number of hepatocytes, with a dose of 50 million TU transducing over 75% of the hepatocytes in the liver. And given the frequency of single and dual color cells, it appears that we have a multiplicity of infection of approximately 2. And importantly, when I injected a set dose into mice and then harvested the livers either 4 days or 14 weeks thereafter, I found that these transduced hepatocytes persisted throughout the lifetime of the animal. And so this was really exciting because it meant that we had a way to stably deliver transgenes into any frequency and fraction of hepatocytes in the liver in an otherwise wild-type animal. And from a screening perspective, what this means is that at a dose of 50 million TU, we transduce approximately 10 million hepatocytes in a single postnatal day 1 mouse liver. And so that equates to about 200-fold coverage of the typical genome scale guide RNA library, and therefore brings us within the ballpark of delivery required to actually perform genome-wide screening in this organism. And so the next question was, can we actually use this system to deplete proteins of interest in the liver? And so to do this, I settled on targeting a long-lived protein MAO-B. And to induce Cas9, I relied on commercially available inducible Cas9 mice, which we can induce Cas9 in all hepatocytes via intraperitoneal injection of an adeno-associated virus expressing CRE. And so I injected a lentivirus with a guide RNA targeting MAO-B into neonatal mice. And then shortly thereafter, I induced Cas9 in hepatocytes with AAV CRE, and then harvested livers at defined time points thereafter. And what you can see is that in the absence of any Cas9 induction, we don't see any ectopic depletion of MAO-B, even in cells expressing the guide. However, when we induce Cas9, we see the specific depletion of this long-lived protein, exclusively in cells expressing the guide RNA. And in neonatal hepatocytes, when the hepatocytes are still somewhat proliferative, this takes about 2 weeks. But importantly, I also tested whether we could deplete a long-lived protein in adult hepatocytes where there's absolutely no turnover. And indeed, we can, although it takes about 1 week longer. And so with this system in hand, we actually wanted to see, can we actually perform a genome-wide screen in the organism? And so we elected to perform a screen for hepatocyte fitness in the neonatal mouse liver. And to do this, we injected a genome-wide guide RNA library into neonatal mice. And then we took half the cohort and simply harvested their livers 4 days after as an induction time point. And in the remaining mice, we induced Cas9 and allowed these mice to grow up for 3 weeks, allowing for approximately 3 population doublings of the hepatocytes. And so at the end of the screen, we then sequenced the guide RNAs in the 4 adult mice at endpoint and compared them to induction. And what you can see is that in a single mouse, we could actually identify both significantly enriched genes, meaning their knockout caused the guide RNAs to expand, and significantly depleted genes, meaning knockout caused those guide RNAs to deplete over time. And importantly, when we looked at these results across the mice at endpoint, we found a really strong correlation between the gene scores across individual mice. And so this means that not only can we technically perform a genome-wide screen in a single mouse liver, we have the power of readily scaling up to perform biological replicates. Now, of course, an important question is, are these results even meaningful? Are they telling us things about cell fitness? And so to do this, we combined the results across all mice, which gave us statistical power to identify more hits. And we then leveraged known human genetics data where we know that certain genes, when knocked out, cause hepatocyte lethality, and other genes, when knocked out, cause enhancement of hepatocyte fitness. And so when we looked to see how these genes performed in our screen, we found that indeed, genes known to be required for hepatocyte viability were depleted in our screen, and genes known to enhance hepatocyte fitness when knocked out were enriched in our screen. And so I'm going to have to end at this point just to show you, though, that we've been able to successfully perform genome-wide screening in the mouse liver, and we can uncover the complete regulation of cellular phenotypes in the organism using this tool. And so to briefly summarize, in this screen for cell fitness, what we were in time able to uncover is that we could find that pathways known to be essential for cell fitness were indeed essential for fitness in hepatocytes in the organism. But beyond this, we were able to find that certain genes actually regulated fitness in a sex-specific manner. We also found that hepatocyte viability was shaped by extracellular factors, including the immune system. And so this tool really allowed us to uncover broader regulation of something as simple as cell fitness that we would have never been able to uncover in cell culture. And so using this tool, we now have a means of performing high throughput functional genomics in the organism that is comprehensive. We can query the entire genome and detect both enrichment and depletion. It's adaptable. We can induce Cas9 and protein depletion at any time point. Provided we have a means of selecting for any given phenotype or staining for it, we can now screen for it using any CRISPR-based approach. And it's accessible. It requires similar reagents to cell culture screens. We can identify hits in a single mouse and is easily scaled. And so I'm so excited about this approach to really bring the power of high throughput functional genomics into the organism and allow us to dissect various organismal processes. For us, we'll use it for regeneration, but I'm really excited to see what other people will be able to use it for. And so with that, I'd like to thank Heather Keyes, who was critical for helping us build this technology. She runs the functional genomics platform here at the Whitehead. My other lab members who are now leveraging this tool to ask questions about liver regeneration, and the other core facilities at the Whitehead Institute who've been incredibly helpful as this project has unfolded. Thank you.
Great, Kristin. Thank you very much for that really nice talk. I'd like to move on now to Abby Sarkar, who's from Stanford University in Roel Nusse's lab. And she will be telling us about assessing the impact of nutrition on epithelial tissue homeostasis and physiology in the liver and beyond. Abby, take it away.
Thank you, Kathy, and thank you to the organizers for this fantastic discussion and workshop. My name is Abby. I'm a postdoc in Roland Nusse's lab at Stanford University. And today I'll be talking about our studies looking at nutrient intake and its impact on epithelial physiology in the liver and beyond. So nutrient intake fluctuates in natural populations. There are times when the organism will feed, and in times when nutrients are not available, an organism will fast. And really, most of our studies in mammalian systems have been done under constant feeding conditions, this ab libitum feeding condition, where the organism is given access to food 24/7. And under this context, which Kristin just really highlighted very well, most cells and tissues are quiescent. So we wondered if we induced a nutrient fluctuation feeding regimen by intermittent fasting, if this would then induce cell proliferation and if this was induced by growth factors. So just to give you our conclusion from the start and tell you how we got there, indeed in hepatocytes, we saw this proliferative expansion. So why do we choose to look at the liver to start with? The liver really is a model organism to study metabolic change. And as Marino highlighted quite beautifully, it's organized in these structures called liver lobules, where there's a portal to central axis. And in between are stacked many hepatocytes, the main cell type in the liver. And hepatocytes are really a heterogeneous metabolic workforce. The reason for that is that blood flows nutrients and oxygen, and this gives hepatocytes very distinct metabolic functions. So hepatocytes near the portal vein, here seen in zone 1, have distinct metabolic functions than those in near the central vein, zone 3, where they receive the end of the blood flow. And I'm highlighting lipid ketogenesis here because it's the main metabolic shift that occurs during intermittent fasting. And this is enriched in zone 3 cells around the central vein. So we really don't have good cell reporters to look at these different metabolic populations in hepatocytes. So single cell RNA sequencing has really helped us here. And using transcripts that we know are specific to zone 3 cells, zone 2 cells, and zone 1 cells, we can then organize these cell populations within our hepatocytes in the liver and calculate their percentages. And this actually highlights something quite interesting. In intermittent fasting, zone 3 hepatocytes are increasing, and they're increasing by almost twofold. So to visualize this increase in the liver, we lucked out in that we had a zone 3 genetic cell lineage tracing system. And so here I'm showing the system in ad libitum conditions, where hepatocytes around the central vein is a good marker here for zone 3. You can see the label, the GFP label, but in intermittent fasted livers, you see this label start to expand. And again, this is about a twofold increase, similar to what we saw in the single cell RNA sequencing studies. So next we wanted to understand how this expansion was occurring, and we were particularly interested in cell-to-cell communication. So given that our expansion is in zone 3 next to the central vein, we thought Wnt signaling may be involved here. The central vein endothelial cells produce a ton of Wnt ligands, and these ligands then go on to bind to receptors in the hepatocyte and induce a signaling cascade. An endpoint target protein here is TBX3, and we've shown in the liver that TBX3 is important in allowing the cell to progress through M phase in the cell cycle. So what happens when we lose TBX3 in intermittent fasting? Here again, I'm showing this expansion of hepatocytes in zone 3 from the central vein in intermittent fasted conditions. And then when we knock out TBX3 in intermittent fasted conditions, you see reduction in this expansion. And because TBX3 is regulating M phase progression in the cell cycle, the cells are rather continuously going through S phase and as such without cytokinesis and mitosis. And you can see this in the polypoization expansion of the nuclei in this model. So that's great, but really what is it about fasting and refeeding that's triggering this proliferative response in the liver? Fasting and refeeding triggers systemic hormones, often many produced in the intestine, and these can go into the bloodstream and a signal to neighboring and far distant organs such as the liver. So endocrine FGF15 signaling was really interesting to us because its receptor, KLB in hepatocytes, is highly abundant. And we wanted to test whether or not FGF15 binding to KLB in hepatocytes would then regulate S phase in this proliferative response in intermittent fasting. So what happens if we remove the receptor in hepatocytes? Again, here we're seeing the expansion during intermittent fasting of zone 3 hepatocytes. And in our knockout KLB receptor mice, this intermittent fasting is, the expansion is not occurring. And because this endocrine signaling pathway is involved in S phase progression, we don't see endoreplication and polypoization of the cells either. So just to summarize the discoveries we've made, first, nutrient intake really impacts hepatocyte proliferation and physiology. And this is coordinated both by local signals in neighboring cells in the liver, as well as systemic signals from other organs. And we've shown that these systemic and local signals work together to really help the cell progress through cell cycle and induce proliferation. So we're interested in seeing if this model is conserved in other organs, but really in thinking about nutritional change and its impact on epithelial physiology as a whole, three main questions come to light and have been highlighted in this conference. And that is how to develop tools to map metabolically distinct cell types. I've shown in the liver that this is really important because hepatocytes are extremely diverse in metabolic function, but this applies to other tissues, and growing evidence is showing that they're metabolically distinct cell types in other tissues. Secondly, the ability to trace cells in real time in response to fasting and refeeding. Here's an image showing from our refeeding that within 30 minutes, we can see proliferative response. So having tools to really trace this physiological change in real time would be of great interest. And finally, identifying approaches to look at cell-to-cell communication in the same organ and to look at cell-to-cell communication within different organs as well is important because these two can coordinate a physiological response. The last point I want to leave us with is that excess nutrient really is associated with the growing prevalence of obesity in the US. So here I'm showing CDC data of the distribution of obesity in the US over the last decade. And as you can see, it's growing. We're at 42.4% now. So really studying the impact of nutrition on epithelial physiology can help us better understand how this disease takes place and other metabolic diseases as well, and design preventative measures and treatments. So with that, I'd like to thank the wonderful lab members, students, and collaborators that have helped me with this project. And I'll leave it to the discussion now.
Okay, thank you very much, Abby. That was really fantastic. And I think I'd like to thank all of the speakers for some very provocative talks today and for summarizing some of the challenges and some of the potential tools that we need to develop to make progress, not only in model systems, but of course, bridging this to mammalian organ systems. So what I would like to do now is hand this over to Prisca. We're going to be handling the discussion by integrating questions from the audience, and we already have some that we're going to start with, and also going to our panelists for their further thoughts. So Prisca, do you want to start with the first umbrella question that you posed to us at the beginning?
Yes, I think that one of the aspects that then came clear also during the talk, one aspect is really the visualization, but also the perturbation of the system, perturbing the system at cross biological scales in a spatial temporally controlled way, and really trying to combine both a cell autonomous type of perturbation and a non-cell autonomous type of perturbation. And I think also the last talk with both local and systemic type of perturbation. And I think I would like to address to Marino one question that brings also some of the question from Patrick Chitwood that we had is, so how can we combine this? And one of the questions we had is, RAP35 seems to be ubiquitously expressed in many issues. So how could this specificity in the morphogenesis that you see of RAP35, and how can we then move forward to understand the role of RAP35, considering that it's ubiquitously expressed, and to understand it in the liver more specific?
Thank you, Prisca. I don't want to say that the RAP35 is obviously liver specific. It is not. We actually took RAP35 as a control because we wanted to demonstrate that our system, which is, by the way, is based on lipid nanoparticle delivering a siRNA. This is basically the system which you are getting now in vaccination for delivering mRNA by BioNTech, Pfizer, and Moderna. So this system has allowed us to basically make some controls. And if you can remove junction protein, of course you lose polarity. We were expecting the same for RAP35, and I think we got actually a loss of this bulkheads, which is not, of course, a knockout for RAP35, is a depletion. I would say that very likely this means that RAP35 is doing something which is key to the formation of the bulkheads. I don't think it's a good idea to remove RAP35 at all, because you will have problem of lethality. In fact, RAP35 removal in various systems is lethal. But I think it is regulating or orchestrating something there with junction, acting, recycling, that is rate limiting for the formation of the bulkheads. And other components will be the same. CDC42 has also a phenotype there. I would say many of the genes identified to be important in hepatocyte polarity should now be revisited because maybe their function is specific for the formation of these structures. You're mute.
I saw it. So if I can continue, Marino, is because there were quite some questions on if you could describe a bit better the morphological. I don't know if Zev also wants to come on, or G2. So both had questions that really address part of really what is the role of actin and how is it really. And if I can take also a question from from the audience, from Cornelia Schweier. So one of the points is, how do the cells sense this? What are the junctions that sense it? So I think the three questions go a bit in the same, and I don't know if Zev or G2 want to comment a bit more on the question.
Maybe thanks, Prisca. Marino, could I, just the idea of these actin scaffolds from inside the cell, contributing to building this tube is very interesting, because it sort of harks back to some of the issues yesterday about intercellular structures organizing the architecture of the cell. But now here you're faced with the structure of the actin, as well as the possibility of volume regulation and mechanics. So I was just, and that may give rise to the larger scale that you see of these pneumatic ordering of these tubules. So I'm just wondering if you could integrate that somewhat.
Yeah, so obviously we don't have much more information than what I showed at this stage. We know that manipulating actin with drugs, different types, they don't have just an effect, a pleiotropic effect. Some of these drugs really can interfere very specifically with this lumen formation, which can, if it doesn't elongate like this, will become globular, because the cell will, if they don't form these narrow tubules between two cells, then what the cells will do, we rearrange, they will move, and then start to form a large cyst. So therefore, the whole notion is that exactly like in a boat, the cells start with live cell imaging, we can see they start making really like a zipper, these connections. And as the lumen elongates, we'll make more of this connection. And we just continue like this until it will zip, and it will zip to the next cell doublet. So that, to us, is an element that is a mechanical element that keeps basically the two cells prevented that they fall apart. And now, of course, with the acting, we have super resolution microscopy, but we don't have really a ultrastructural reconstruction of the acting. But what we see by super resolution microscopy, we see really the acting going into this projection, and then probably we would imagine they meet then the junction at the end. So we will believe that these are under tension in this way. So that the junction prevent them from falling apart, and the acting, of course, will emanate as filaments coming from the tip of those ridges when they connect there. So there is obviously acting throughout the tube, acting as every other apical surface is very enriched, but is particularly enriched in dominating really in this projections between the two cells. We don't know more than this, unfortunately, at this stage.
So Marino, just to add a little, so is there the system, the network on the tension, and is the flow of bile inducing something to stabilize the network?
So the flow, that's an important question, because I think, part of the aspect I think that we are dealing with is in some system, you have fluid dynamics that will govern how the cells really are, when they proliferate, etc., or when they modify, how they get together. So obviously these cells pump fluid into the apical surface. If they were not having the bulkhead, then it will form a cyst. So they have to pump fluid for elongating the lumen. And of course, there is an increase in volume because of course the lumen it starts as a small globular lumen between two cells, and then it extends really by making this. So it has to increase in volume, but the volume increase has to be anisotropic again, exactly because we find that these are this structural element that prevent the cells really from going other way.
Thank you very much, Marino. I don't know if...
Should we go on to a few questions for Abby? There are a lot of questions for Marino that are still in the chat, and hopefully we'll capture all of those. But we'd like to get through a few more questions for other people. So there were a couple for Abby, both in the area of this first question, but also in tools. So I'll save tools for later. But Daniel Feliciano has a question about intermittent fasting affecting overall zonation. Daniel, do you want to elaborate on this question? Can we bring you in? I'm not sure.
Yeah, I can take a stab at it. So it does change zonation in the liver. And as I highlighted, it's really increasing zone 3 and decreasing zone 2. So that's what we're observing.
Great. And then there is, here I see Daniel. Do you have any other follow-up to that?
Yeah, that was pretty much my question, to know if that increase in zone 3 was on the expense of another zone, right? Because every zone has a role. So I just wanted to know if that was the case. Thank you.
And then Solanga Nakul, I don't know if I'm pronouncing that correctly. Is the fasting effect through hormones or lack of nutrients reaching the liver through enterohepatic circulation?
Yeah, so we focused on hormones in the talk, but it would be really interesting to do individual nutrient tests and nutrient sensing in the liver as well.
Okay, great. Prisca, I don't know, do you see other questions for Abby right now?
No, I think that we can, I think Jennifer had her hand raised for a while.
Yeah, so my question was similar to Daniel's about whether some cells were being absorbed while others are being proliferated during the starvation. But related to that, I'd actually like to hear from Marino in terms of, or both Abby and Marino, in terms of the cells that are proliferating under fasting. Are they sort of driving the sort of flow and the dynamics of the system that Marino's talking about, or are they shifting the system more to, for instance, mitochondrial oxidative phosphorylation, in which case you'd shut down that flow system?
Yeah, so this is a really interesting question, something that I'm really excited about. We completely ignored the biokinetically, the sinusoidal network in this talk. And I would be really interested to see how it's changing with intermittent fasting. And in terms of cellular structures like mitochondria and ribosomes, there have been studies that have shown that high-fat diet, excess nutrient intake, changes those structures quite dramatically using EM imaging. So I would suspect the same thing is happening in intermittent fasting.
Have you looked, Marino, at all at fasted liver with your beautiful reconstructions?
Yeah, I think that actually there is a lot of interesting things happening in the fasting liver or liver, which is, also there are things which occur short term and the things which occur long term. And very interesting is also, maybe it's beyond a little bit this topic here, but a little bit the problem of liver diseases, really. What happens when cells start to accumulate lipid droplets and become fatty liver, foie gras? And there are, there is a very, almost like a deterministic series of pathological changes which occur in the tissue, where metabolic changes result in alteration of tissue structure. And the alteration of tissue structure results in cell fate changes. So it's a lot of interesting things going on, which reflect a little bit of the discussion yesterday. Yeah. And I think again show that how much really we have to think about new concept about that the cell and the tissue are not just determined and they stay forever like that, but they change a lot depending on diet, on other injury. And then they can take a path that we can lead them very far away into repair and then making wrong decisions. Because the liver disease really from metabolic disease that they have, they end up having a fibrotic tissue, a non-functional tissue, and at the end producing cancer. So it's a very stereotype pattern. And I think nobody knows really in that field what happens.
Thank you. Really exciting.
Yeah. So just to go back, Marino, also on what was said yesterday, there were some more questions by Zev Gardner and Thomas Lequi about the difference between a cyst and a tube and about the structure forming like at the corset to stabilize the tube. So can you speculate, can you discuss a bit about it a little more?
But my understanding of this problem, I mean, there are beautiful reviews actually written in the field by various people. I'm not going to cite anybody, but I think it's clear that you must have some anisotropy in at the level of whole tissue in order to elongate, or otherwise the minimal energy would be to produce always a sphere, right? So of course in the tissue there are tensions that are causing the cell to reorient, and then the tube really a tube really forming and avoiding forming a cyst, when it has to avoid it. So anyway, at the scale that we work on is just a 2-cell doublet, but at the scale of a whole epithelium, there must be similar mechanisms that would have to put tensions really on the whole tissue. And something like this is what Jennifer was talking about, right? You have forces at the level of the tissue that tell cells what they have to do. And of course, cells will respond to them and generate new forces as well. Maybe Jennifer, you could comment on this.
Yeah, I think that'd be great, Jen. Would like to hear from you on that.
Yeah, it's certainly possible that mechanical forces could coordinate a lot of these different behaviors in cells. And we're really at the tip of the iceberg, I think, in terms of understanding how cells respond to forces and what are the in principle, forces could stretch any protein that's anchored somewhere in the cell. So any protein that's anchored to the nuclear membrane or to the plasma membrane in two places, or to the cortex, would be stretched when those structures are stretched. So I think it'll be really interesting to start looking more at what kinds of proteins can be stretched under tension if we can develop tools to look at that in living cells.
Yeah, so we can, I think, then move, Jennifer, on the tool. We had some question by Alan Kay, but I think generally about different tools. So if there are reliable GFP-based force sensors, and I think we can integrate both one aspect on what do actually cells sense and respond to. Is it stiffness and elasticity? But then which tools can then we use to then understand how cells sense mechanical environment to then respond to them?
Right. So I think developing those tools will be an important challenge. So, I mean, right now, for example, we use myosin 2 localization to as a readout of where forces are, because myosin generates contractile forces, and it also is it responds to forces, but it's not perfect because it's not a tension sensor. It's just a protein going to where it needs to go in the cell, but it also happens to respond to tension. So really, I think we need better force sensors that actually we can tell are measuring force, and better ways to actually measure forces.
And to go back, do you think halo-based tools might be really good? So there's one question from Marco on Strabo that, do you think halo-based tools could be useful?
Yeah, I don't know too much about them. So I think we need to explore a variety of different approaches.
Mm-hmm.
Jen, one of the things I was wondering, also it's something that I think about a lot, that I like to hear more about, is this idea that we really need to come up with ways of looking at very rapid changes and post-translational modifications that are occurring, multiple ones at the same time that are occurring at real time, both in homeostatic conditions, but also under external force or other kinds of stimuli. And I'm just wondering if we might get into a little discussion on that. We talked about it a little bit yesterday about nanobody libraries, aptamers, things like that. What are your thoughts on that?
Yeah, I think the idea of screening nanobody libraries for nanobodies that can be genetically encoded that can bind to specific post-translational modifications, or maybe epitopes that are normally buried but are only exposed when a protein is stretched. I think that kind of thing would be awesome.
Yeah.
So I think there are some hand raised. So Michael Caplan, you're there since a while.
Thanks, Prisca. So these were 4 wonderful talks, and I have several questions for each of them, but let me ask a question to Kristin, and this maybe reflects my ignorance, but you have developed this exquisite technique for performing this in vivo mutagenesis. I'm wondering how one screens for the sorts of phenotypes that we're talking about here. In other words, phenotypes that relate to cell-specific architecture or even tissue-level architecture, as opposed to things that affect proliferation or viability.
Yeah, I think this is a great question, and something I think we have some tools we can tackle these problems with now, and then I think there's ways to think about how we could move on beyond this. So as a proof of concept, we did a very simple fitness screen where we're looking for changes in abundance in the bulk population. But because we have the ability to dissociate single hepatocytes from the tissue, that means provided there's a way of staining for the phenotype of interest, you could in principle then sort for it by flow cytometry or laser capture, and then sequence guide RNAs in the phenotypic high population and the phenotypic low. Now that then restricts you to a phenotype that is something you can stain for, whether it be an RNA or a protein or a given expression of a protein. And so I think we can get pretty far with those types of phenotypes, but that still really limits us to cell autonomous phenotypes and expression-based phenotypes. And so we had been chatting actually as a group last week about, can we go further? And so I think one thing that's true is that because we deliver our guides in the neonate, there's going to be some clonal expansion. And so you'll actually have a few hepatocytes in a patch that will be knocked out for any given gene. And so you could imagine doing image-based screens where you look to see how has the morphology of that patch changed. Now, an image-based screen at genome scale might be at present infeasible, but this method you could apply to any sort of library. So if you wanted to go scale down the library to a set of targeted genes, you could then deliver that, and then your patches would be a bit more defined in terms of what they are knocked out for. And by imaging, you could look at the morphology of those patches and then identify the guides in those populations. And so I think this is really a framework for something that we can really build and adapt to a bunch of different things. It's ready to go for single cell phenotypes that we can stain for, but I really hope that we can actually push it to these interaction-based phenotypes and sort of tissue level phenotypes next.
Kristin, do you think you could that it would be possible to apply like single cell RNA-seq to look at different cell types that are depleted for certain guide RNAs? So maybe those guide RNAs are required or knocking out genes that are required for the differentiation of that cell type?
Sure. So we could. So I think that in terms of the screening system we developed, it's hepatocyte specific. So we induce Cas9 specifically in hepatocytes, and to our best knowledge, the guide RNA lentiviral delivery targets hepatocytes specifically in the liver. Now we're working on ways to broaden that coverage to other cell types. That said, within hepatocytes, there's a lot of functional heterogeneity, as has been acknowledged in terms of where they are on the axis. And so you could imagine a perturb-seeked approach where you inject the genome-wide library, do a genome-wide mutagenesis screen, and then isolate hepatocytes and look at single-cell RNA-seq to correlate knockout with transcriptional changes. And all of that would be possible using this method.
So there was a question early on for Kristin from Elaine Fuchs, but I have a feeling she may have left. Do we have Elaine with us?
I think she left. Yeah,
left.
she had to leave.
We'll get that question to Kristin and she can answer Elaine separately. But I know we also have several hands up. Prisca, do you want to?
Yes, so there's, I think, Praps, I think was there, and then Sylvie.
Hi, great talks. I had a question for Marino about those bulkheads that you described. I was thinking, is there any, like, do you know if there's a diffusion barrier for proteins and lipids between that structure and the actual bulk membrane? And if it's there, then probably does that mean, if there's no diffusion barrier, does that mean that membranes of adjacent cells are actually continuous? And also I was thinking, like, do you think this is kind of might be a general structure when you want to actually create tubules, like for example, the intestinal epithelium? Are structures like these have been basically observed in other like physiological tissues?
Yeah, so we don't know whether there is really a barrier there in those structures. We see, of course, a continuous membrane. Apical markers are seems to be going into the structure too. So, but we have to look for more markers than that. So, and also, you know, at the end, we have to understand better really the structure of that system, in the sense that to really see what is special about the junction, and is there anything else that would make that place really that projections into the lumen to be special. Now, concerning other structure, I'm not aware of other structures like this, in the sense that I'm not aware of two cells that are immediately juxtaposed and form this kind of structure. To some extent, of course, I would say that these are adaptation of projections that cells in general can do, but adapted in order to just make a sort of a zipper between two adjacent cells. But I'm thinking, for example, some of the structure that you see in the kidney, in the glomeruli, right, when they are this podocyte that are really, you know, forming projection like this, they're not engineered to be between two cells. They could, of course, if they had junctions and things like this. So I'm saying the cell in the hepatocyte exploit pretty much the same machinery to make projections, but adapt it in order to make sure that two cells juxtaposed negotiate their apical surfaces so that they meet with each other all the time, really. I should also say that this pattern, I forgot to say, this pattern is periodic. There is a pattern. It's almost like the same distance between each of the bulkheads, like exactly you would make it in a boat when you build a boat. So I think that there is some way of measuring the acting and making sure that this acting really maybe nucleates this projection at a sort of regular distance. But of course, mechanistically, we don't know what's.
Thank you, Marino. We have a few more minutes left, so I wanted to move Sylvie Breton. You have a question? If
Yes,
it's
thank
Marino,
you.
If it's Marino, short answer, short question, short answers.
Well, I want to bring back the notion of the question about how cells know to form tubules versus cysts. And I think it's a fascinating biology question that we are wrestling with. So my question might be either for Jennifer or Marino, whoever wants to answer. But my comment is actually, we have found also similar tubule structures in vitro by epididymal cell lines that were not subjected to any mechanical stress. We just put them in a Matrigel, and I was really surprised to see them actually forming nice tubes and also branching these tubes. And so there was no polarity in stress except for gravity, perhaps. But what was interesting to us is that by knocking down CFTR, we actually found these cells do not know to form tubes anymore, but they just form masses of completely dysregulated tumors, basically. So it seems to me that the mechanical force may be one factor in an organ system, but in vitro, without these directional mechanical forces, we can still see those elongation of tubes. So what would be your interpretation on that? I've been wrestling with this myself.
Jennifer, you want to go?
No, Marino, I think you should take this one.
Okay, so I see what you mean. I mean, for example, sinusoidal endothelial cell from the liver as other endothelial cells, they form tubes. So they can form tube in matrix or other kind of, so it's clear that they don't need to have a tension or pressure like in the tissue really to do that. But they must still, I mean, there must still be fluid going into these things, otherwise they will not form a lumen because they have to. That's why I would say the interpretation of the CFTR is that there are no ions going there, and they are not going to, you know, to have osmotic pressure to occur. So you must have pressure anyway, and then the cells maybe they have to divide or they have to align themselves in order to extend it. But it doesn't mean that in vitro you have to have the same tension or forces that you have in the tissue. So it's still a cell autonomous and local cell interaction problem. We can continue later because.
And can I add to that one thing? Matrix is very important for this process. I would suspect if you put these in alginate without any biologic adhesive properties, they would just grow a cyst. So I think it is the combination of fluid in a tube rather than just a cyst.
Thank you for bringing it up. Absolutely.
So I think for the last minute, there's something to go back on for Abby. Is like, how can we move forward to be able to then measure and to go back to metabolism, different metabolic state in a dynamical fashion, in full tissue, both combining this local and more distal type of interaction and coordination between cells?
Yeah, it's a major challenge, one that we discussed a lot yesterday. And I think there's there are ideas that are coming forward that emphasize the need for scale, using model systems, and then scaling up from that to what's going on in a tissue as a whole and how different tissues interact. And in terms of technology, that's evolving as well. The use of nanoprobes and different reporter molecules, glycans as potential tools that we can start using it for this technology.
I don't know.
Great. I think we're almost at time, 43 seconds.
So I will just then, I think, conclude for today's session. And I think that what came out is quite important is to understand complex shape and function that really goes hand in hand. So spatial organization and functional. We will really need to cross biological scales to be able to visualize and perturb. And I think that the development as a community of very quantitative method to measure and measure beyond gene expression, so signaling mechanics and metabolism, and to have this combined with specific perturbation that can bring us and to like combine our understanding to go back to this complex shape and function that really, and feedbacks that then are really very important. And with this, I think I would like to thank all the speakers. So Jennifer, Marino, Kristin, and Abby, this was a great session, and looking forward to the rest of the day.
Thank you, everyone.
Thanks, Prisca and Kathy, and thanks to all the speakers. I agree, it was a fabulous session and a wonderful way to start today. And at this point, I'll turn it over to Patrick, who's going to introduce the next session.
Okay, great. Yes, the next session is titled Innovating Informative Model Systems. And we have three speakers. First will be James Wells, from a professor at Cincinnati's Children's Hospital. Next will be Ora Weisz at University of Pittsburgh, and Adam Edwinson, who's a postdoc at the Grover Lab at the Mayo Clinic. And then our discussion will be led by Janelia's Carolyn Ott and our own executive director, Ron Vale. And I'm looking forward to a great session. Let's take it away with James.
All right. Well, thank you. Can you see my screen?
Mm-hmm. Yes.
All right. All right. Well, this has been a fantastic meeting. I don't need to tell you all that. So I'll just go right into my talk. I will thank the folks in my lab and my fantastic collaborators and funding sources. And the focus of this session really is on emerging new model systems to study physiology from the cell up to the organ. And the subject will be organoids. And organoids, I'll talk about later, have been generated from or as a model of many different cell types and organ cell types shown here. And a lot of the excitement around human organoids has been modeling human disease, which is, of course, a meritorious and important thing to do. However, as a basic scientist, I get just as excited about being able to model cell and tissue physiology using organoids. And the general system that we've been focusing on are the organs of the GI tract. Now, the GI tract, as you've already heard a lot about, is essential, is the center of nutrient homeostasis systemically. Not only does it regulate the ingestion, digestion, and absorption of nutrients, it also communicates with other organs throughout the body to control things like satiety, as you've already heard, and nutrient mobilization by inter-organ communication. Now, at the organ level, it's extraordinarily complex. It's comprised of cells from all three developmental germ layers. And through interactions of the epithelia, which is on the inside of the GI tract, with its neighboring layers of smooth muscle, neurons, immune cells, vasculature, it coordinates a lot of the GI functions, again, ranging from mechanical digestion to absorption. Now, the epithelium, which is, of course, one reason why we're all here, helps to coordinate a lot of the organ and systemic functions by interacting with adjacent smooth muscle and neurons, as well as interacting with distant organs, as we've just heard from Abby's talk, by secreting hormones that can, of course, act systemically. So organoids of the GI tract are being used to model, at the moment, mostly a lot of the cell and tissue physiology. And organoids, just in general, predominantly come from two sources. They can come from human surgical biopsies. Again, I'll talk really just about human organoids today. So primary tissue-derived organoids can be perpetuated in culture more or less indefinitely. And GI organoids from biopsies, this technology really was pioneered by Hans Clevers and his colleagues. The other type of organoids, which my lab has predominantly focused on, although not exclusively, these come from the directed differentiation of human pluripotent stem cells in vitro to de novo generate organoids. Now, these types of organoids both have their strengths and weaknesses, but in general, they've really allowed us to study human biology in lots of new and exciting ways. And one reason for this is that you can reconfigure organoids into any variety of tissue shapes and sizes. So you can put an organoid in 2D, as shown in the upper left here, where you can study nutrient absorption as controlled by enteroendocrine cells. If you want to look at morphogenesis or slightly more complicated aspects of tissue biology, you can grow organoids in 3 dimensions, although this has its drawbacks as well. Or you can engineer more complex 3-dimensional systems. Shown below here is an intestinal organoid. After an additional 12 weeks of growth in an animal, you can see villus morphogenesis. And in the right here is a movie showing engineered neurons aligning with smooth muscle in this human intestinal organoid. In the future, however, though, it should be possible to generate systems that allow for inter-organ communication. And I'll discuss a little bit of this at the end. Now, even in the simplest forms, these organoids can have some of the basic properties of the epithelial tissues. So you can have barrier function, you can secrete acids as in the stomach, you can absorb nutrients and secrete hormones, and these epithelia have their characteristics of the different organs. And at the moment, they've mostly been used very effectively, I'll add, to model human congenital disorders, infectious diseases. They're really quite good for because many pathogens are species restricted. They're scalable, so you can do biochemistry and omics. You can do gene editing, and of course, in vitro, you can study some of these processes using live imaging. So some examples of how we use these organoids in vitro or to model simple processes are shown here. On the left, we've used a laser-induced injury of a single cell in an organoid to show the cellular and tissue dynamics of epithelial injury and reestablishment of barrier integrity. And this is just on a loop. This is all epifluorescence and just staining nuclei. And you can see a nice dynamic response of how the epithelia push this cell out and reestablish integrity. On the right here is an organoid system we developed recently where we have resident macrophages in green responding to an inflammatory cue and how it interfaces with the colonic organoid shown in red. And you can very nicely see in real time, macrophages become migratory, they secrete cytokines, and they induce apoptosis. So there's some utility here, but we're of course really keen on building better systems. So for example, for epithelial injury, we've now developed an E-cadherin M-Ruby fusion shown on the right here that co-localizes with endogenous beta-catenin to show that we will use to look at dynamics of epithelial injury response and how cells and cadherin complexes break down and reform during this whole process. So again, this can all be done imaging in vitro. But as I mentioned, I'm really excited to use this as a basic discovery tool. So we recently were able to use organoids to connect how a nutrient-absorbing enteroendocrine cell actually signals to its neighboring enterocytes to control the absorption of carbohydrates and proteins. And went on then to confirm that in a mouse model of malabsorption to show that a single hormone could in part rescue a malabsorptive phenotype in this mouse model. And again, this was actually all based on a human mutation that causes malabsorption. So again, as a basic discovery tool, I think they can be quite useful. But I do want to point out, this was really technically challenging. Organoids had to be grown and then deconstructed into 2D, and as I said, disassembly required in this case. But there are new organoid systems coming along which allow for more user-friendly access. And I'm going to highlight one right here that came out of Matthias Lutoff's lab recently, where they generated in vitro using sort of an engineering-based approach mini gut tube. And you can see these loops here are actually crypt-like domains, and the lumen is actually highlighted in the middle here. And what they've been able to do is actually allow luminal access to this organoid while not perturbing the system. So you can see in just a second, you're going to see the luminal access, whoosh. I had to add the sound effects there because Matthias didn't have that in his movie. But you can see now there's luminal flow. And this system really brilliantly was used to study homeostasis of the epithelium for months, injury, regenerative responses, as well as parasitic infections. So I think there are coming online new systems to study the various aspects of the epithelia of the GI tract. But what about more complex organ function that require more than just the epithelium? So my lab's developed an approach where we can now at will build added complexity into organoid systems by adding to the epithelial component of any given organ, smooth muscle or enteric neurons. And this is done all starting from human pluripotent stem cells. We can combine these various progenitors in very restricted amounts, and they will actually self-organize to give rise to somewhat functional tissue. So our first proof of that was actually generating enteric neurons in an organoid system. On the left here, you see enteric neuron precursors that were just added to an organoid a few weeks prior to this video, efflux and calcium using the G-Camp line developed at Janelia. And this IPS line was genetically generated in Bruce Conklin's lab. But importantly, those added neurons can then go on to control smooth muscle contractility in this human organoid-derived tissue shown here. And this contraction, this peristaltic-like contraction, is in fact coordinated by the neurons that we engineered into that tissue. Now, what about the next level, inter-organ communication? The gut, as has already been nicely discussed, both controls nutrient absorption and digestion, but it also signals around the body to control things like food intake, satiety, glycogen storage in the liver, and insulin secretion. So how can we use these sorts of systems to study inter-organ communication? And this really is where biomedical engineering has come in very remarkably useful, in that engineers can develop fluidic platforms onto which we can generate cell and tissue constructs and study inter-organ communication. And this is just one example of a project that is in my lab with my collaborators around the country, Muyil, Taka, and Eben Alsberg, where we're gearing up to show that this system can both study nutrient absorption in the intestine, but also how the nutrients absorbed can then signal to distant organs like the liver, as we just heard, perhaps through FGF signaling, or the endocrine pancreas. And we can, at will, put these organs in series or in various combinations to look at inter-organ communication in vitro using this fluidic platform. And this is all in development, but that is our hope. And there have been some really nice proof of principle studies. So what I would like to see in moving forward, of course, we've already discussed the absolute need for better reporters and biosensors to look at things dynamically. So it was mentioned, wouldn't it be great if we could monitor a cell secreting something and then another cell receiving that signal in real time? Absolute need to study cell metabolic pathways in real time, to look at how cell and tissues change their cellular metabolism in response to various insults. And then, as discussed, better quantifiable morphometry to look at cell and tissue size, shape, and volume. Better culture, better culture methods to get at organoid functions in real time. I mentioned one out of Matthias Lutoff's lab. There are a number of labs looking to generate microfluidic devices that have vascular beds into which organoids can grow. So we would potentially be able to regulate the input through the vasculature and then monitor the output through also the vasculature. And then really generating bioengineered systems to study these processes. And again, why do organoids make cysts instead of tubes? This is a great, potentially ideal system to study what controls that, because we can manipulate physical and genetic inputs. We can manipulate flow, stretch, inducible gene expressions and whatnot. And then lastly, of course, at will to engineer added complexity to really then take the functionality to the next step. I already thanked the folks in my lab. I want to thank the pluripotent stem cell facility, really, which generate all of our genetic tools and a lot of the processes. And I'll stop and we'll talk about this at the discussion session.
Thank you, James, for a very good talk. We can now move on to our next talk, which will be Ora Weisz.
Hi, I want to thank you for the opportunity to present my work and to participate in this amazing workshop. So we haven't heard a lot about the kidney structure or function yet, and so I'm going to spend a couple minutes of my precious time just introducing you to this amazing organ. So the function of the kidney is to maintain blood homeostasis, and in order to do this, it filters about 200 liters of plasma every day, and out of this it recovers about 199 liters of water, salt, metabolites, sugar, etc., and creates a liter of forming urine. And in order to do this, the kidney is made up of up to 1 million nephrons in humans, arranged in this kind of parallel complicated array. And there's up to 20 different cell types that have been described in this structure. So one of the unique features about the nephron is they start with the glomerulus, which is a filtration device where the plasma is basically pushed from these capillary loops into the tubule lumen at fairly high pressure. And then the rest of the nephron functions basically as a beautiful Rube Goldberg kind of machine where the ion transport in individual segments creates the driving force that enables subsequent segments in the nephron to take up other ions. And a couple things that I want to point out about the kidney are, first of all, that you can see that the interstitial ion composition changes dramatically as you move from the cortex into the medulla. And we actually have cells in our body that can withstand 1,200 milliosmoles, for example, in external fluid. Another feature is that the structure of these nephrons differs depending on their positional localization. So cortical nephrons that are up near where the glomerulus is up near the outside of the kidney capsule have a somewhat different structure than juxtamedullary nephrons where the glomerulus is placed closer to the outer medullary junction. And we still don't understand much about how these different structures relate to kidney function. So some of the big open questions that I think need to be addressed in the kidney field are, how do nephrons acutely adjust their ion transport and also their endocytic uptake of filtered proteins in response to normal changes in flow, such as the ones that occur after you drink a cup of coffee or finish your burger? What are the cues that drive cell differentiation across along the nephron axis? Is it nature or is it nurture? Similarly, how do cells adapt to these incredible microenvironments in different regions of the kidney? And conversely, how does the microenvironment influence cell function? Also, how does nephron segment identity adapt to physiologic changes? So for example, during pregnancy, it's been demonstrated in the rat that the proximal tubule grows by 20% during pregnancy and lactation and then reverts back to normal. Is this a change in cell size? Is this a change in cell division, et cetera? We really don't know. How does metabolism affect kidney disease progression? And so, all of our cells are bathed in serum, which has 40 milligrams per mil of albumin, which can carry some toxic substances. But why is it that when a little bit of protein leaks into the tubule lumen, that all of a sudden the kidney goes haywire, as in under proteinuric conditions? And then how do cells within nephrons and between nephrons communicate? So there's evidence for synchronization between nephrons. There's evidence for nanotubes being able to deliver substances to cells along the tubule lumen. And there's also evidence for exosome-dependent communication between tubular cells. So what are the things that we need to measure to answer these questions, where there's the sort of classic 4D cellular physiology things like metabolic changes, mitochondrial dynamics, membrane traffic, ion transport, and pH and ion composition, cell-cell communication, flow-dependent responses in mechanosignaling, which we've heard about already. And ideally, we'd love to be able to do this in intact animals where you can preserve this incredible structural complexity. But there's a lot of challenges to doing this in vivo, particularly when you have a living animal with a heartbeat and a breath to take care of. So it's very difficult under those conditions to get subcellular resolution to measure some of these things. Because of the glomerular filtration barrier, we can't easily control or measure the flow rate or the protein composition that enters the tubule lumen. And as this image that Janos Peti-Peterdi graciously shared with me, there's a lot of regional heterogeneity in the kidney in regards to blood flow, metabolism, and transport. This just shows you albumin uptake in grayscale along the kidney. And you can see that there are regions that take up a lot of albumin and some that take up less. Similarly, you can't really visualize membrane traffic or follow protein fate in real time in these. And there are also some strain and sex-dependent variations in kidney structure, as well as organism-dependent variations that make the relevance of some of the animal models to human disease a little bit suspect. So I posit that we really need to use a wide variety of different levels of resolution to understand kidney function, going from cell culture, organoids, model organisms, microdissected tubules that can be perfused, deep tissue imaging, and then clear tissue, among others, as a way to really try to understand kidney function from the molecular level to the systems level. And by way of disclosure, I'm a classically trained cell biologist, so my preference is to start with cell culture models and then work my way up to the kidney. You'll hear later from other people who start the other way around, and I think that mix is a really nice balance in our field. One of the problems with using cell culture, however, is that kidney cells are highly morphologically adapted to their function. And these are some images from Dennis Brown's beautiful work showing you just some of the incredible morphologic specializations that cells that line different regions of the kidney nephron maintain and are required to maintain in order to really fulfill their function. So my laboratory studies endocytosis in the kidney proximal tubule. It's an extremely endocytically active cell type. And basically what happens is you have, again, 40 mg per mil of albumin coming into these glomerular loops. Most of it is excluded from the tubule lumen, but a small amount of it gets in. And then this albumin binds to receptors at the apical surface of proximal tubule cells and is readily internalized and degraded, leaving essentially no filtered protein entering the tubule lumen. And so in order to study how the cells adapt to these, generate and maintain this incredible apical endocytic apparatus, we really need to be able to look in model systems. So this is an EM of a mouse kidney proximal tubule, and you can see how incredibly complex the apical endocytic pathway is. There's tons of different kinds of tubules and vesicles that form this structure. And so we can't use a generic kidney cell like an MDCK cell, for example, which is great for studies of membrane traffic, but not necessarily good for studies on endocytic, the endocytic pathway in the kidney proximal tubule. So we've optimized an available cell model that unfortunately comes from an opossum. The opossum kidney cells are OK cells. And under our culture conditions, we can nicely replicate this subapical complexity of the apical endocytic apparatus in proximal tubule cells. And if you look more closely, you can see again, both at the apical surface and at the basolateral surface, we can approximate the morphological specializations that make proximal tubule cells what they really are. So how do we do this? Well, unfortunately, it's not possible to get this using the standard closed system microfluidic setups that are commercially available, because the two cues that you need in order to generate this kind of differentiation are continuous oxygenation and mechanosensitive cues. And so the way that we've been able to do this is to culture our cells on transwell filters and then just put them on a rotating shaker in the incubator. So there's some huge advantages and also some big disadvantages in using this approach. However, using it, we've been able to answer a lot of questions about what the organization of the apical pathway looks like, and also to look at its responses to changes in normal variations in flow. And this is just to show you that we found that the endocytic capacity of these cells can change acutely and very rapidly and reversibly when you adjust flow rates in these cells. So we think that this may be one way that cells adapt endocytically to normal variations after you have a cup of coffee, for example. But we can't really tell easily whether this kind of adaptation also happens in vivo. Again, because we have no way to deliver known amounts of ligand at a known flow rate into the tubule lumen. But I will tell you that we can't just use these cell types in order to develop these systems, because when you bust open the glomerular filtration barrier, the proximal tubule is able to dramatically increase its capacity to internalize filtered proteins. And it's been estimated that there's about a 60-fold increase in its endocytic capacity under those conditions. And that's something that we haven't been able to replicate in vitro yet. And probably the reason why comes from a study by Andrew Hall, where he injected a fluorescent filtered protein and a fluid phase ligand, dextran, into circulation of mice, and then looked in cleared kidneys at uptake of these two probes. And you can see here that on the left, the filtered protein gets taken up very rapidly after it leaves the glomerulus, and but it's only after its uptake is almost complete that you start to see appreciable increase in uptake of this fluid phase ligand, dextran. And I should remind you that the proximal tubule recovers about 70% of the filtered water, both by paracellular and transcellular mechanisms. And so I would posit that under nephrotic conditions where you're overloading the receptor-mediated uptake of fluid ligands, that now you're increasingly taking up increasingly concentrated albumin and other filtered proteins by fluid phase uptake in more distal parts of this nephron segment. And again, this is something that we can potentially test in vivo. So what are the things that we need? What's my wish list for being able to dissect nephron function? Well, I'd love to see more highly differentiated primary or immortalized rodent or human cell lines that retain these specific nephron segment functions. Most of the cell lines that we have are sort of generated serendipitously, and I think that concerted efforts are really tried to establish Such cell lines would be hugely beneficial. It would be lovely to have co-culture models that would allow us to resurrect nephron segmentation and allow the interaction with vascular or immune cells. And also we need to be able to mimic these in vivo interstitial compositions that we see in the kidney. It would be lovely to be able to rapidly manipulate the microenvironment, such as tubular flow and basolateral stretch of these cells, while also being able to image these at high resolution. And I, for one, would love to have a way to image apical endocytic uptake while acutely manipulating flow.
Or I just wanted to give you a quick time cue.
Okay,
sure
this is my last slide. We need scalable models that can enable us to do drug screening and gene knockouts to look at and look at various output measures. It's been mentioned many times before. We'd love to have optimized probes to quantify ion concentrations and look at endocytic uptake. Enhancements to deep tissue imaging that would allow us to look at superficial versus juxtamedullary nephrons. And of course, computational tools that would let us take information from all of these multiple systems and put them into a comprehensive model for kidney function. So thank you very much.
Okay, great. Thank you for that wonderful introduction to the kidney, which we're going to hear more about a lot in the next session, I think. So this is going to be a perfect segue. But for our last talk, let's get to Adam Edwinson. Go ahead, Adam.
We up and running?
Yep,
All
looks
right.
good.
Thank you. So just want to take a quick moment to say thank you to Janelia and the organizers for inviting me here today to give a talk on some of the work that we've done on patient human-derived colonic organoids and the responses to the enteric pathogen Campylobacter. So I just want to give a brief introduction on the disease that we study in our lab. It's called post-infection irritable bowel syndrome. With regards to this, we see that annually there's about 48 million US adults that suffer from a case of infectious enteritis. And acute infectious gastroenteritis is commonly identified as a risk factor for the development of IBS. When we look worldwide, we see that the prevalence of post-infection IBS, or for the sake of this presentation, we'll call it PI IBS, ranges anywhere between 4% and 36%, with nearly 14% of bacterial enteritis cases resulting in IBS development alone. We focus primarily, though, on the United States. We see that a major driver of the development of PI IBS is, in fact, bacterial infections, which leads us to why we study Campylobacter. Campylobacter, being a bacteria, is one of the most common causes of bacterial enteritis annually, with approximately 10% of Campylobacter cases resulting in PIBS development. One of the problems that we have is that our understanding of host pathogen interactions in the context of Campylobacter and hosts and really enteric pathogens in general remain really understudied. And when it comes to these studies, it's important to recognize that the intestinal epithelium is incredibly complex, one, but it's made even more complex in the context of PIBS, as changes can be seen in epithelial biology throughout the entire GI tract of these patients. We see that there can be bile acid malabsorption, increased 5-HT secretion. We've ourselves have published data on decreased microbiome diversity, as well as an increased presence of proteases in the lumen and in the presence of pro-inflammatory cytokines. We also know that there's a decreased expression of tight junction proteins, which inevitably leads to increased permeability. We see specifically decreases in ZO1, occludin, and claudins. We also see that in some patients that there's an increased presence of immune cells, whether it be mast cells and macrophages, and actually in some instances a shift in the Th1 to Th2 ratio. And finally, we see that some patients have neuronal excitation leading to visceral hypersensitivity, and in some instances neuronal depletion. So you can see that there's a lot of heterogeneity in how PIBS presents itself. So with regards to our understanding of PIBS development, there are certainly some gaps that still remain. To this day, typically we've used immortalized cell lines to study these host pathogen interactions. These are nice because they help reduce confounders and keep our systems clean, and they're homogeneous. However, that also comes with a drawback in that it limits our understanding of host-specific effects. As I had mentioned previously, Campylobacter host pathogen interactions remain relatively understudied, and that's largely in part due to the fact that conventional mice are resistant to colonization of this bacteria. So they don't present the disease phenotype that we would associate in our mouse models. Also, germ-free mice, which have been used, they fail to recapitulate any potential microbiome interactions that may be occurring within the lumen of the intestine. And finally, as I had mentioned above, we are missing addressing some key human elements. We know that females are more likely to develop PIBS compared to males, and that anxiety and depression are often associated with increased risk of PIBS development. So we decided that we would take a chance and take a look at how patient-derived organoids could be used for modeling infection. So what we did is we recruited a total of 20 patients and divided them evenly among high stress and low stress phenotype. We were able to generate organoids from 15 out of 20 of those patients, so about 75%, of which 8 were generated from high stress, 7 from low stress. And we plated these organoids on collagen-coated substrates for experimentation. And we had to do this because scanning the literature, a majority of studies have used substrates such as Matrigel, but one of the issues with using these types of substrates is lots of times they're embedded with some type of antibiotic. And in this instance, Matrigel is embedded with gentamicin, which is a drug that is used to treat Campylobacter. So we had to look at alternative substrates for this. And finally, we looked at 2 different clinical strains of Campylobacter jejuni and assessed the barrier function, transcriptomic and proteomic profiling, as well as did some imaging and the infectivity of the bacterium itself. So one thing that in our lab that we find is incredibly helpful and innovative in measuring our barrier integrity of our organoids and cells in general is the use of this CellScope system. The CellScope system provides automated cell monitoring of our monolayers. We can take real-time recordings of transepithelial resistance to study barrier function. And the nice thing is that this system here can be incubated, so therefore we can measure cells under physiological conditions. And it allows us to measure multiple wells at a given time. We find that this is superior to handheld TER measurements because it provides both automated measurements as well as stability in the electrodes. There's no variation that you would experience with something like chopsticks. We also, this system has a Teflon block at which the trans walls are loaded into. So the Teflon block holds heat. So when we remove this system from our incubator, we have limited concern with regards to cells cooling when we add any substrates or bacteria. And a nice part about the system is that we can add and collect from the apical and basolateral chambers without removing any liquid or disturbing those said electrodes. So what we found was that, as to be expected, Campylobacter being a bacteria can cause barrier disruption in these organoids. I just want to highlight here both high stress and low stress organoids that we cultured are able to form stable barrier, and this stable barrier lasts throughout a long duration of time. I have this time course out to 36 hours. However, we've had these up to 70 plus hours in this CellScope system. Now, looking at these strains, strain 9 and strain 26, as I said, we used 2 strains. We can see that over time, that there is a distinct loss of barrier function in the presence of, or the addition of the bacteria. However, what I want to highlight here is that in our high stress organoids, we see a relatively similar pattern based off of the strain. Whereas in our low stress organoids, we actually see an uptick, an increase in barrier function during the initial time course of the infection, at least in strain 9, not nearly as pronounced in strain after exposure to strain 26. But we also see that it drops down as to be expected. So I wanted to pull a specific time point from those tracings to demonstrate what is going on here. So the barrier integrity is obviously being affected by these this bacteria, but it's also interesting to look at the phenotype. So we've specifically focused on the high stress individual patient organoids. We see that approximately 50% of the initial TER remains after 24 hours, which is significantly different from our control organoids held under the same conditions, however, not being infected. We look at this low stress patient organoids, we see that barrier function still remains fairly high after 24 hours, anywhere between 60% with strain 26 and then at about 80% for strain 9. So we see this differential response based on the stress phenotype. So we took this a step further and we wanted to look at some tight junction proteins and see how they were affected. And we found that they were damaged during infection. When we look at the high stress organoids, we see that there's a nice clear staining of occlusion highlighting the junctions between our cells on our trans walls. Whereas after exposure to both strain 9 and strain 26, more so strain 9, there's a complete obliteration of occlusion protein staining within the junctions. And there's a slight degradation in the strain 26, but not nearly as much with strain 9. We see a similar visual with regards to the low stress individuals, nice staining of occlusion in our control uninfected wells, but not nearly as much degradation at matched time points. We follow this by co-staining with DAPI to ensure that yes, indeed, those are cells. And then we also coupled that with phalloidin staining to highlight cytoskeletal elements of our cells. So RNA transcriptomics, we found that they reveal strain-specific epithelial responses to the infections. I want to highlight first that when we look at stressed and non-stressed organoids that have not been infected, so these uninfected controls, we see that there are 17 differential pathways that are predicted to be different, but all of them relate to metabolism. So it was nice to see that we're not seeing baseline differences in like immune markers. So when we compare strain 9 versus strain 26, stress versus non-stress individuals, we see that actually in strain 9, there were 124 gene sets that were enriched in organoids from stress individuals, where only 7 were enriched in after exposure to strain 26. And I want to highlight that of the top 5, 3 of which were immune-related after exposure to strain 9, whereas only 1 was found in strain 26. So some of the limitations in our approach: intestinal organoids, they help recapitulate human condition. However, Campylobacter is microaerophilic, surviving best at low oxygen concentrations. And to date, the experiments that we've conducted have been in an aerobic incubator. The effects of Campylobacter, therefore, on the epithelia and the permeability may not be actually fully realized. We also, in these systems, lack contributions of the microbiome and immune cells to the overall response of the invading bacteria. Also, these studies have been done on monolayers, and these do a good job at studying the XY planes. However, we're limited in our understanding of how Campylobacter and enteric pathogens in general behave in the context of a three-dimensional crypt. We lack a complete understanding of the effects on cellular architecture, barrier function, and permeability, as well as pathogen behavior in a three-dimensional space. So where do I see the need?
Just a quick time cue here.
Beautiful, thank you. Should be the last slide. Where I see this research going and where we should push our resources toward to innovate this research is really keying in on what Jim had brought up, is getting closer and closer to mimicking the luminal environment. So these organoids are close, but what I suggest is if we can develop a system where we have a completely enclosed 2-chamber system, where we have apical media feeding the apical surface, basolateral media feeding the basolateral surface, and we can even add the enteric pathogens or the microbiome to the apical surface and feed it with physiologically relevant oxygen concentrations, whereas the basolateral surface is at 95%. So in this system, I would like it that we would be able to image while also taking electrical resistance measurements at the same time, so we can hit 2 birds with 1 stone. And finally, collecting the apical and basolateral effluents. And beyond that, thinking in a 3-dimensional space, there has been progress in this area, but it would be nice if we could look towards imprinting membrane supports that at which organoids could adhere to. So we seed our organoids in the basement of these imprinted membranes so that the organoids can grow up, form these nice curves to which we can do experiments in. Very quickly acknowledging the ENSP group at the Mayo Clinic, as well as the funding sources, because I couldn't do this all by myself. And I appreciate your time today.
All right, thank you. I'd just like to thank all 3 speakers for great talks. And I think now I can hand it over to Carolyn and Ron for our discussion.
Terrific. Well, what I hope that you can notice in these 3 talks as a little set is that there's kind of a contrast between the strength and the potential of the organoids and the complexity of what's happening in a tissue. So Aura, explains so beautifully all of the complexities in the kidney. And with Adam also kind of pulling out for us the diversity with the bacteria and with the samples and the origin of the samples. So there's a lot for us to unpack in terms of how do we get tools and models that are going to really give us information. I think Ron's going to start us off with some questions from the audience.
Yes, I'd like to get the audience involved in our global audience involved first, if I could. And I'm not sure if I can get a spotlight, but Mira Krendel had an interesting question. There are a couple of questions for Aura, and I think they're on the theme of, how do we mimic what might be complicated tissue functions in simpler systems, which I think is a theme of this session overall. Oh, Mira, there you are. Okay, yep. Wonderful. Why don't you go ahead and ask your question, please?
Hi, so I thought it was a wonderful talk and really highlights the complexity of the kidney. And what I was thinking about, even when we look at the kidney sort of in vivo or in the cryosection, it's often difficult to decide what are the good markers to use for the different segments of the nephron. And if you're trying to reproduce it in vitro, how do you determine whether you were successful? And so would you be looking more at the physiological readouts that you mentioned, how responsive they are? Or would we need to go back to something like single cell transcriptomics and try to figure out what are the appropriate markers for the different cell types? So how do you envision that working?
Oh, it's a great question. I mean, we do have actually some really good single cell data from the kidney, from Caplan, Sustak, and Ben Humphrey's groups, among others. And, it's clear that there's so many different cell types in the kidney. So I guess that, for me, I've always used functional readouts to try to get at this question in terms of looking at what are the key roles of these segments, and then what's the expression level of the key molecules, so that you don't necessarily look at the entire transcriptome, but you look at a segment of it related to the function that you're interested in looking at. And then also, how does that change when you put the cells or allow the cells to grow in an environment that more mimics their natural microenvironment? So, there's no really good answer to this, right? I mean, you hope that you can establish, it's a chicken and egg question. Does the microenvironment establish the transcriptome, or does the transcriptome establish the microenvironment? And so how do you get this thing going? I don't know.
Great. Thank you, Mira. Juanita also, Juanita Merchant, I don't know if you're there, but I think Juanita was also commenting on kind of an unusual environment of the kidney. We talked a lot about mechanical forces driving morphogenesis and cell fate, et cetera. That's been a common theme. But Juanita brought up the fact that, the kidney also works in an osmotic gradient. And that is a little bit of an unusual situation to compare to other organ systems. And I don't know, Juanita, if you're there, but anyway, I'll continue with your question.
I don't know what happened to see. Okay.
Oh, there you are. Okay, thank you, Juanita. Okay, I got you started. Anyway, keep going.
Yeah, that's fine. I just wanted to bring up the whole issue of the gradient. It also will probably pertain, because I work in the stomach too, this osmotic, the concentration and composition of the fluid. So it's not only pressure, but it's going to be the composition. And the stomach obviously is very acidic, but then there in the kidney, which is impressive, you've got, the cellular, the changes in the differentiation and phenotype of the cells are probably impacted by the concentration, the salt concentration in the kidney. And that's going to be hard to mimic. And I don't know whether that's been able to be reproduced in organoids or even some of these cell types, culture cells.
No, but that's a place where the engineering folks at Janelia could actually have an impact. I mean, I'm sure that there are some either bioprinted or sponge-like materials that could help to sequester particular ions and help concentrate them so that as you're driving differentiated function, you can accumulate the driving force that you need to really stimulate cell differentiation. So, but I don't know, I don't know of any systems out there that are done. I think that's one of the limitations of kidney organoids, frankly, is that you just don't have that kind of difference in spatial accumulation of osmolality.
Microfluidics could maybe also be used to set that up too, which is, I know, bioengineering, biomedical engineering situation.
I think it has been done in the intestine, which also, does a lot of coupling to ion gradients for both water transport and nutrient transport, as it turns out. But I think Adam's sketch at the end of his of these sort of simple fluidic systems that really are just two-chambered, I think you can actually set it up that way, obviously, and establish gradients and monitor electrochemically how gradients are affecting tissue function. Again, I've seen it in the intestine. I haven't necessarily seen it in the kidney, but I would imagine it's possible.
Thank you.
Great. Thanks very much, Juanita. I see lots of people have their hands up. Prisca, Catherine, I do see you, so I'll get to you next. I want to pass things back to Carolyn for a second, though, because even those last questions brought up issues of how we go back and forth comparing whatever we do in a in vitro model system back to, the reality check of the complexity of the tissue. Anyway, I think Carolyn wanted to pose a general question on that. So go ahead, Carolyn.
And I think this kind of plays off a little bit what Mira was asking and Aura answered, but kind of taking it another step further, like what do you need to lower the barrier, to make it easier to go between systems? Because we're looking at, the individual cultured cell system as kind of a reductionist and the organoid as kind of an intermediate, and then the organism. Are there some, fiducials? Are there things that would make it faster to go between these so you can really validate the physiology of what you're looking at? And I think any of you, maybe start with Jim and then Adam and then Aura kind of fill in.
Yeah, at the moment, no. So what I mean by that is pretty much everything that we've discovered in an organoid, we've pretty much had to go back and recapitulate in an animal model or even in a patient before people would believe it. So I think we're at the stage now where, you still have to validate with whatever you choose to benchmark it to, and that can be kidney function in a mouse or a patient biopsy to recapitulate a phenotype that you first discovered in an organoid. Ultimately, I'd like to think that things will be benchmarked and become uniform enough where the technologies can be used in multiple labs with the same results at the end. And I think that's been the biggest challenge for the organoid field, is they're still really technically challenging to get working in the lab, and transferring that to another lab, even a lab that does this for a living, can be challenging. So there will always, in the immediate future, we'll still need to benchmark to something else. So like for us, if we build complexity into a system, we have to then do a single cell analysis of the new system and compare it with actual human fetal colon or what have you. So ultimately, I'd like to think that won't be necessary, but it's still important, I think, to benchmark to something that you trust, a mouse model or another cell line, whatever.
There's multiple dimensions of benchmarking, both the organismal, tissue level, as well as across spaces. Adam, can you pull from your experience?
Yeah, I would have to agree with what Jim had said. We're at a point right now, all things considered, organoids are still kind of new, right? And we're trying to get them to a point where everything is reproducible and trustworthy. And at least in my experience dealing with human patient organoids, is that 75% success is pretty good. There have been some labs that hover around 20%. And I think what would help, at least in my experience, is when I first started the project, is that scouring the literature. You can do, as Jim said, try to replicate one lab's protocol and get wildly different results. And if you look from one paper to the next, they have different culturing conditions. They have different levels of certain metabolites. So I think what might help is a standardization of some of these types of things. I know there are certain companies that have begun to commercialize the organoid media. So trying to make things as standard as possible will help reduce some of the issues that we currently have in trying to make that next leap. But I also think, there's a flip side. We've talked a lot about, the other side of the lumen, the neurons, the muscle, etc. There's also another organ, the microbiome, and trying to balance that, understanding how these organs as a host are interacting with each other, but there's also a major microbiome component at play here. So balancing both of those is, I think, a major thing that we need to reconcile as moving forward.
There's so much in that. That's terrific. Aura, do you want to just add a little bit, and then we'll get to some of the hands that are up?
Sure. I mean, I think, I've always been astonished that you can take this ball of cells and when you make kidney organoids and they can actually segment into, different nephron segments, a little bit rudimentary, but still, even in the absence of flow, you can get this kind of differentiation going. And so one of the questions that I actually sort of have is, if you want to try to establish culture conditions to generate cell lines that really mimic function, do you start with organoids? It's a matter of scale that might be an issue, or do you start with immortalized cell lines and change the fluid? But is there some way that you can sort of take advantage of that intrinsic microenvironment that organoids are able to self-generate in a way to try to create uniform lines that can be used from the cell to the animal?
And maybe before we go on, I want to honor like our worldwide audience, or in some cases Janelia audience, but just pick up on with what Adam said right now, because I think I can summarize two questions that came in from Francisca Dengler and Andy Lemire. And that is, you take the organoid, in your case, it's out of a patient, and the patient, one of the questions is, it's a hypoxic environment. It also may be, for that individual, a stressful environment, and that's how you partitioned your two groups. You must have done this before COVID-19, because it's now hard to find a low stress cohort these days. But anyway, you take this out of a patient, and then you put it in culture. And the question is, like, how in that transition time are you retaining that some of the elements of the natural tissue environment, like the stress of low hypoxia, whatever physiological stresses were happening for that patient? How well do you think that transfer is happening from the patient, to the culture system?
I'd say it would be irresponsible to suggest that it's 100% the same, but there have been studies that have shown that when you culture organoids from tissues, that they retain the phenotype from which they came. So I would certainly concede that there is a potential for change, but what we have done is we've gone back and looked at patient biopsies and compared their transcriptomic profiles to organoid monolayers that we've cultured, and we're not seeing drastic shifts in either differential genes or pathways that are differential between our organoid versus our patient biopsy. So I guess with regards to that maybe addresses that question. With regards to the hypoxic environment, I think that's been I think that's a sticking point for a lot of GI researchers, because a majority of our studies have been in cell culture, not necessarily held under hypoxic conditions. So I think that's where I would like to see the research go is that we're getting more innovative. If we start bringing in the engineers, the biomedical engineers, to develop these systems that can make our relatively simple experiments a tier more complex, we can start to answer these more specific questions revolving around differences in oxygen concentration and really get at recapitulating the environment.
Okay, great. Well, now I'm going to turn to our very patient larger panelist group, and I would say the chat session is often really interesting to read, so I encourage anyone to add to that as well. But I think Prisca had her hand up first, so I would like to pass things on to you, please.
Yes, thank you for really for the great talks. So I have a question that mainly is for Jim, but I think it's for a bit everybody. It's like, so the majority of these organoids and self-organizing systems generate really local microenvironment, and this is the power of organoid. They can self-organize not only their signaling niches, but also like extracellular matrix. So what do you think are the major challenges to really combine this self-organized, the local environment of the cell with engineer environment, both of matrices and fluxes that then tend to recapitulate the more in vivo? So where do you see the major challenges at the interface between the self-organized and the engineered?
Yeah, well, I think the challenges are really just designing the systems that you can do both. I mean, I think we've already done, as you've said, the first step of controlling the microenvironment. So which cells will be introduced to one another at the right time and place for them to self-organize into some structure with some level of functionality and architecture. But then, yeah, you're right. The next step is where we're going and where the field needs to go. How do you introduce the next level of complexity or interfacing? And that can be mechanical. It can be, how do you introduce stretch and flow? It can be neurochemical, which requires added cell types to transmit signals, or artificially inducing these through like optogenetics, things like that. But it really is the next step where we can integrate these sort of primitive organoid systems into a device, a structure, an environment where we can then control, for example, the environmental cues. And again, that can be stretch, flow, and whatnot. I mean, that's one approach, if that was the question. If
yes,
not, you can.
Yeah, but also how we can then integrate these dynamical aspects, because how, like, while they self-organize and change and dynamically change internally, how this can be then introduced in an engineered microfluidics that also change over time to respond to cues.
Yeah. Well, you know, that is a great question. Dynamic interfaces are hard to come by. I mean, it would be great if we could have, you know, different types of matrices incorporated into just even represent changing from a sphere to a tube, for example, constraining, you know, geometry of organoids. And a lot of these are, I guess it really depends on the question. We're still at one question and design an entirely new platform to address that one biologic question. We're sort of at that state, getting to the state where it's a dynamic, you know, where you can control the shape of the organoid, you know, say into a tube, and then the stretch to induce smooth muscle, and then the monitoring electrochemical gradients across the epithelia at the same time. That's where I really wanted to highlight to Ron and Carolyn, where biomedical engineering and engineering is really going to be essential to do that. Doing it in a dynamic way, maybe we can chat again in 5 years.
Yeah, in a way to introduce a sensing device that sense the environment, sense the also then feedback and change over time.
Yeah. No, it's yeah. That's the next generation.
Which it's great to have in our conversation, both the near and the far horizon, that we can think about where we're going to be able to go with this. Why don't we go ahead and bring on Michael and then Jennifer to learn from the questions that they have as well.
Could I just say, just briefly, because we have 10 minutes and we can't cover everything, if anyone has any crazy wild bioengineering ideas, just add them into the chat box, because there's all kinds of things about stretch, fluid flow, interesting things. So if anyone has a wild idea that we should be unconstrained and add it to the chat, we'll think about it. Okay, but anyway, yeah, sorry, Carolyn had, yeah, Mike, go ahead.
Thanks. Wonderful talk. So, Aura, I'm curious, and I'm sure you know where this curiosity comes from, as to how you think the shear stress is sensed by cells. And have you ever tried to perturb the function of cilia in your model system or in vivo to see if changes in at least ciliary bending could change the transduction of the shear stress into changes in endocytosis?
So this is actually a case where, having a really a system that really replicates in vivo is more helpful. So we initially did studies like that on cells cultured in EBD chambers, where the cells are basically grown under static conditions, but they develop some brush border and a cilium. And under those conditions, perturbing the cilium seemed to have an impact on endocytic uptake. But in our more differentiated cell lines, we're not convinced that the cilium is really the mechanosensor. And it actually makes sense if you think about the proximal tubule. It has thousands of microvilli and one primary cilium that's just a little bit longer. So if you're going to design a mechanosensor, you probably are going to pick the brush border. I mean, just a small change in the bending of those microvilli could really, integrated over thousands, can really have a much bigger effect on cellular function. And in fact, that's been proposed before by Chang Wang and Alan Weinstein in some beautiful studies that suggest that these might regulate changes in ion transport. So how you test that, we would love to be able to find a way to perturb microvillar structure and ask, especially acutely, ask whether it changes our response to flow. Again, if anyone has any flyers, please let me know.
Thanks. Jennifer, would you like to?
Sure. Yeah. So I have a question for James. I was really excited when you talked about hooking up different organoids with each other, like make an organoid from a liver or make an organoid from a kidney and hook them up through some sort of microfluidic device. I'd like to hear more in terms of how you think that might work, but also whether you could interrogate differences between not just sort of fluid flow between these different organs, but also lymphatics, blood, and nerves, which are the major conveyors that communicate between these different organs. So I think this is so exciting if you can actually hook some of these organs together. So yeah, tell us more about it.
Well, this is a part of a sort of emerging area actually funded by NIH, this large consortium of labs trying to grow cell and organ tissues in vitro and ultimately connect them. I think your point, and I think there have been some good proof of principle fluidic connections that suggest you can get one cell talking to an adjacent organ cell through a fluidic device. And I think there are some very nice publications. The next level of, for example, being able to feed in an input through a pseudovasculature and then read the output out the other end is also, I think, making progress in terms of either through artificial vasculature like fluidics or real vascularity where there are in vitro vascular beds that actually both feed an organoid and you can measure what comes out the other side. So there's some really nice work being done there as well in a variety of different labs. I can send you to references offline. Neuronal connections, to my knowledge, has not been really convincingly shown, where you can actually have a neural, something equivalent to peripheral neurons or even vagal inputs into an organoid system that can actually control their function and feed back. To my knowledge, that's not been replicated in vitro. The best I know of are actually probably CNS communications where interneurons from one regional organoid, actually send projections to, for example, a cerebral organoid in an adjacent chamber, showing that sort of thing. To my knowledge, peripheral innervation and control of a target. Actually, I guess neuromuscular has been done using organoids, where you can get peripheral neuromuscular connections between a smooth muscle or a skeletal muscle. But doing it in a large scale fluidic device, again, very primitive stages.
Cool.
So
thank you.
Terrific. Marino, you've had your hand up for a little while. And
Yeah,
then
so I don't have a crazy idea, but maybe I have a sort of anachronistic idea in the sense that does it make sense to think, I mean, we have a lot of progress with organoids. So we are thinking about extracellular matrix, growth factors. So what about going back to the embryonic organ and culture them in vitro and have them to develop? For example, the kidney, you have the embryonic kidney, you have the ureter bud that goes into the mesenchyme, it differentiates the cells, and then they start forming the part of the nephrons. So how much can you sustain this in vitro? Therefore, my idea is really, what about going back also to the old, now that we have a lot of organoid biology, going back now to the experiment done 20, 30 years ago, and now redo them and support the organ growth ex vivo.
It's a good idea, Marino. It gets really limited by oxygenation.
But that's why maybe microfluidic would help if you can pump, if you can force some fluid going in by some means.
Well, I'm happy to address that. Being an embryologist myself, much of what we've done, in fact, is in part recapitulating, as you said, experiments done 20 years ago. And those are what inform a lot of what we do now. So we're redoing experiments now to take mesenchyme from one organoid, put it on another organoid to ask, does it change that organoid's fate? So yeah, I think that's a fantastic idea. My former postdoc, Jason Spence, basically he figured out how to generate human lung organoids from stem cells by actually working with human fetal lung tissue. First, to identify what are the factors, matrices, cocktails that are required for outgrowth of human fetal progenitors, and then applied that to deriving organoids. So I think the field is wildly populated by developmental biologists who are doing exactly what you suggest, really taking it to the basic principles of organogenesis in the embryo.
And could I just ask a brief question about that, just to follow up? Because there's a question from the audience, from Isabel from Janelia. And there are different paths to make an organoid. One is, you mentioned iPS cells, mixing them together. Obviously, development is more complicated. There, if series of maturation events, which gives rise to an adult organ and the organoid that you derive that from. So how similar are the organoids that you derive from a completely synthetic model? I think this is the basis of her question, versus one that comes out of natural development. And this also integrates with Marino, what Marino just said. Maybe you can give a brief answer to that, because I think Thomas also has a question.
Yeah, we now routinely benchmark the organoids that we make in vitro with actual tissue at the single cell level. The biggest differences in organoids grown in vitro are metabolic, and this was discussed yesterday, that if you grow something in vitro, it's got a lot of metabolic aspects that don't exist in vitro. So it uses different energy as a food source a lot of times in vitro than it would in vivo. The biggest differences are, I think, because they're in culture. However, there are, of course, many overlapping similarities with regards to what cell types do you get in vitro versus what actually exists in vivo. Those benchmarkings show that, as you would expect, the cells that you engineer in vitro do have counterparts in the actual organ, but you really have to be careful when comparing an in vitro derived organ with something that is a fetal tissue. So single cell benchmarking now is the gold standard.
Lightning rod question from Tomas, with 30 seconds on the clock.
Yeah, I think it's a very quick question. Sorry, maybe it's very naive, but about sustaining growth of an organoid, what about making something hybrid where you would have a mouse and you would sort of bypass its vascular system, taking advantage of the self-organization of vascular system? If you graft a limb bud into an embryo, there's a new vascularization that just grows naturally. So if there was a way to sort of connect, vascular system of an embryo as it grows, or even a young adult, connected to an organoid, so you would sustain the growth of the organoid. Doesn't sound to me like science fiction. Has it been tried?
We do that routinely. Actually, that's a great question. I didn't really talk about it. We use an animal's blood supply to grow these organoids to tissues that are as big as your pinky. So that's a great question. And in these Blitz talks, you can't really go into the details, but that's exactly what we do now. It'd be nice that we wouldn't, we basically call it our sort of in vivo incubator.
So that's the future for sustaining growth in vitro then?
Yeah, well, mimicking an actual blood supply in vitro, as it turns out, is some heavy lifting, but there are labs attempting to do that.
Okay, great. I think that marks a pretty good end to a great session. And so I'd just like to thank all of our panelists and our speakers. And we have a 10-minute break to rush down and get as much food in as you can before we start again at the next session. So we'll see everybody in a bit.
All right, I think now would be a good time for us to get started with the second half of today's talks. And I am very pleased to be introducing session 7, which is entitled The Challenge of Studying Epithelial Cells in Their Natural Habitats. As far as our speakers, we have David Bryant, who is a senior lecturer at the Beatson Institute of the University of Glasgow. We also have Michael Caplan, professor at Yale and co-organizer of today's workshop. And we have Seham Ebrahim, research fellow at the National Cancer Institute of the NIH in the lab of Roberto Weigert. Today's discussion will be led by Denise Montel, professor at UC Santa Barbara, and Wallace Marshall, professor at UC San Francisco. So with that, let's get started with Dave Bryant.
Okay. Can you see the presentation?
We can see it.
Okay, perfect. Thanks very much for the invitation to give my musings on the future.
Actually, Dave, I'm sorry, I'm going to interrupt one time. We see it, but we're seeing your presenter mode. So
fixed.
There you go. Perfect.
Yep. Okay. Thanks very much for the chance to chat. My name is Dave Bryant, and I'm at the Cancer Research UK Beatson Institute in Scotland. And I wanted to be very cheeky in the first instance and say the subtitle for 4D cellular physiology maybe needs a little bit of rework. Instead of saying exploring how cells work in a dish, work in tissues and not a dish, maybe what we actually need to do is build better systems that reflect how cells work in a tissue without throwing out the dish. And if we're in the mood for editing, maybe I can edit myself and say, what I think we need to do is really serve up better dishes. So what's the dish we have the problem with? Well, I think the problem is this concept of 2D culture. But 2D culture is a really powerful tool. It's taught us a lot about what we know of how cells interact together, and really is a stalwart of understanding of cellular biology. And on the new expanded menu, where Hans Clevers initially was the lead chef, I think we've seen 3D culture grow in the last 10 years. And this 3D culture can be achieved through many means, as we heard from Jim Wells, but invariably involves plating either on top of or inside matrix, either cells or cell lines or fragments of tissues. These will undergo proliferation and self-organization to form structures containing a lumen if they're epithelial, and proliferation in the plane of the monolayer, as well as secretion into the lumen, can form more mature structures. But even simple cell lines such as MDCK that I've indicated here, which is a K9 epithelial cell line, can be used to understand how collections of cells organize their polarities centrally around a single central lumen. And this is perhaps best understood by using time-lapse microscopy to understand the dynamics over time. And so I spent a number of years of my life trying to understand in this basic system how you build a single central lumen between cells, including how the cells recognize the ECM and how apical basal polarity is built de novo where it doesn't exist before, including the trafficking pathways that deliver proteins to the lumen, as well as the fail-safe mechanisms based on apoptosis that can occur when these mechanisms are inhibited. And what this led to was really a big list, a list of proteins, either cargoes or trafficking molecules that regulate transport to the lumen. But I think after making this list for so many years, one of the most striking findings was not any identity of any single molecule, but really the key lesson that we learn over and over is that most of these phenotypes do not manifest in 2D culture. Most of the molecules that we've indicated here do not have defects in apical polarity when generated in 2D culture, even from the same cells. So 3D, even if in a dish, does have some of the magic dust required to make cells organize collectively into mimics of tissues, albeit these are always a reductional model. And so what I wanted to have a musing on was what did mapping apical transport pathways for so many years actually lead to? Did this lead to anything more than understanding a cell line in vitro culture? Well, I'm going to give a few vignettes, and these are not to exclude the many fantastic works from many labs, but these were some of the papers that I thought were really impactful on myself and our lab's research. And the first is work from Marta Shabazi's work at Cambridge, England, not Cambridge, Massachusetts, that showed that many of the cargoes and transport machinery identified in these simple 3D culture models in vitro were shared during embryogenesis in mouse and human embryos. And in fact, the transcription factors that regulate exit from naive pluripotency actually control the transcription of both the machinery and the cargo proteins that are required for formation of the luminal cavity during early embryo formation. And that more importantly, inhibition of these trafficking and cargo molecules were intimately linked to the differentiation status of the state, where if lumen formation was inhibited, you actually inhibited egress out of naive pluripotency. And so in my lab, we've been really interested in understanding how do we move on from simple models in 3D and use models that can be transplanted in vivo to really bridge this in vitro to in vivo gap. And what I wanted to point to you, and this has already been very excellently laid out by Jim Wells, is that we still need to ask the question if organoids are now a Michelin star dish or if they're just a new kind of TV dinner. And so there were 3 papers that have really had an impact on me. And the first was one of the early works of the Watanabe lab in collaboration with Hans Clevers that showed that organoids grown ex vivo could be grafted in vivo and restore barrier function to damaged intestine in the mouse. More recently, Nicola Valeri's lab in London showed that patient-derived organoids in co-clinical trials with patients that the organoids are derived from very faithfully mimic the response of these patients to chemotherapeutic intervention. And this is in stark contrast to the last 30 years of 2D cell lines being very poor at predicting these outcomes. And more recently, from the Valier lab, they've shown that ex vivo grown organoids from cholangiocytes can be grafted back in vivo and restore function to damaged biliary ducts. And what's the killer experiment in this is that this completely rescues survival after an otherwise fatal toxic administration to these. And so I think what's also important is that it was very exciting to see last week, actually, Hans Clevers tweet out tantalizing bits of information about the first in-man clinical trial for transplantation of oral cavity organoids to patients that have had oral cavity difficulties post-radiation. So this sounds great, but do organoids really perform like what's on the new wrapper of this TV dinner? And what's the downside to them? So I'm going to point to something super simple, which is a movie that Andy Ewald and I took many years ago where we had MDCK and we just imaged them by phase contrast microscopy. All of these cells are exactly the same in the same conditions plated at the same time. And what you'll see here is the formation of a luminal structure like you'd expect from most MDCK. But I want to point out to you are all of these other phenotypes that are occurring in parallel. And what we mapped was that many of the organoid cultures or cell line cultures that we look at actually display vastly different phenotypes in parallel. And we could identify that there was an inverted polarity in this system that had defects in ECM signaling. And actually, instead of forming a lumen, the apical membrane was driving multicellular invasion. Now, this was heretical, apical membrane-driven invasion, but Fanny Zallen's lab went on to show that actually inverted apical polarity underpins metastasis in specific types of colorectal carcinomas. So this really makes us ask a question when we talk about 3D culture: which of these is the phenotype? If they occur in parallel, how do we know which is the one that we should be studying? These are theoretically the same cells, yet the same molecules are either promoting formation of normal polarity in a lumen or driving collective invasion. Now, the relative ratio of these two in the same culture is going to give us vastly different results, let alone not having a particularly strong way to identify how each of these could be characterized morphologically, let alone have some way to understand their ratio. So we have to ask a question: is what's happening most frequently the most important, or are there outliers, overlooked entities that maybe are the most important? And what my lab has been focused on in the last number of years is finding that actually this heterogeneity occurs across most things that we've looked at. And when we look in particularly tumor samples, we have a common but low frequency event of this inverted polarity. And these are the ones that metastasize from tumor cells, not those with a normal type polarity. So I think what Janelia can do is help us prepare better dishes in our cell biology kitchen. Where we have morphological heterogeneity, I think Janelia can help us with massive parallel multi-day imaging to really understand what phenotypes are occurring in parallel, and allow us with software solutions to try to track these objects and extract features. These might be size, shape, or movement, or biosensors like we've detected or talked about before, and really to define behavior states. Now, Prisca Liberali, who's here on the panel as well, has really been at the forefront of leading the charge to understand different behavior states in organoids. But I think what Janelia can help with is putting these in the context of time. There are computational algorithms that might predict this, but we find that these very poorly act to understand different morphogenesis trajectories. And if we have this, we could then selectively label distinct behaviors, characterize them in vitro, and then translate them in vivo. And so I think the Janelia challenge might be to link genotype to phenotypes, not phenotype. And this would allow us to do cell biology in a dish. And for instance, might allow us to do CRISPR in 3D for the first time, linking genotype to phenotypes. And so the challenge for heterogeneity is to create systems for in vitro cell biology that are compatible with transplantation. Now, for my very last couple of moments, what I wanted to highlight is something that's appeared a little bit over the last couple of days, which is unlocking the challenge of equality. We as a field rarely consider sex differences in our tools. Did most of you know that MDCK are actually female dog cells? And so the challenge I push back on Janelia and for the discussion is how can we embed sex differences in the 4D cellular physiology program? Now I'll just stop here and thank my lab. This is a photo of us as our last Christmas party. This is a photo of me trying to be Mrs. Claus, but the lab ensures me I'm more like Krampus instead of Mrs. Claus. I'll stop there and thank you.
Thank you, Dave. That was a great talk. You definitely shared some really nice ideas for serving up better dishes. So with that, let's move on to our second speaker, Michael Caplan.
So can everybody see my screen?
We can see it.
Okay.
We see your presenter view right now.
Yep, hang on one second.
There you
Okay,
go.
hopefully that's better.
Yep.
All right. Thank you very much, Dave. That was a great talk and actually leads into a number of the concepts I'm going to talk about. So far, we've been talking a lot about the cell biologic aspects of epithelial cells. But I think it's also important to understand what they do physiologically. And a lot of what they do physiologically is transport small molecules, ions, fluids, nutrients from one body fluid compartment to another. And in that context, what a number of the speakers have already talked about, apical basolateral polarity, is really required in order to mediate vectorial transport of fluid and electrolytes. So here is sort of a cell biological view of epithelial polarity with an apical and a basolateral domain composed of different plasma membrane proteins. But when a physiologist looks at an epithelial cell, this is what they see. They see a distribution of ion transport proteins. And if you'll indulge me for a minute, I'd like to take you through how critically important polarity is for determining the physiological properties of an epithelium. What we're looking at here is a schematic diagram of an epithelium found in the kidney, the thick ascending limb, where a basolateral sodium-potassium ATPase drives sodium out of the cell, allowing sodium to flow down its gradient from the apical side into the cytoplasm through a sodium-potassium 2-chloride cotransporter. The potassium recycles to continue to drive this through an apical potassium channel. The chloride leaks out the basolateral side through an electrogenic chloride channel. If you take these same types of transporters and shuffle them around a little bit, leaving the sodium pump where it is, but moving the NAK2Cl cotransporter basolateral and the chloride transporter apical potassium channel basolateral, now you have a secretory epithelium, such as the secretory epithelial cells in the airway or in the crypts of the small intestine. The names of some of these transporters are different, but the activities are the same. And it emphasizes the point that you can switch an absorptive to a secretory epithelium by changing the polarized distribution of membrane transport proteins. And that, of course, raises the question of how can we figure out how polarized membrane transport proteins know where they're supposed to go? Essentially, all of the membrane proteins in epithelial cells are, of course, made in the endoplasmic reticulum. From the endoplasmic reticulum, they travel to the trans-Golgi network. And when I first entered this field back in the mid-1980s, at that time, Kai Siemens and his colleagues had shown definitively that the trans-Golgi network was a major sorting station. And it was thought that there were essentially two destinations, apical and basolateral, and all trafficking decisions were made at the level of the TGN. Proteins, newly synthesized proteins, were loaded into apically directed or basolaterally directed carriers. But as time goes by, I noticed that two things happen. One is that my hair gets grayer, and two is that more arrows proliferate on my slides. So as more and more research in this area was done, it was noticed that there are many more termini in the trafficking pathways from the TGN, including common recycling endosome, apical recycling endosome, apical sorting endosome, basolateral sorting endosome, and recycling pathways from the cell surface to all of these. So we wondered how we could watch newly synthesized membrane proteins traffic to the cell surface in real time. This is the work from who used the SNAP tag to watch newly synthesized sodium pump travel from the trans-Golgi network to the basolateral plasma membrane over time after releasing a 20-degree block. What I hope you can see is that the sodium pump showed up after about 20 minutes or so all over the lateral plasma membrane. But interestingly, when Glenn looked at another basolaterally directed protein, the VSVG or vesicular stomatitis virus G protein, he accumulated them in the trans-Golgi network, they superposed, but when he released the temperature block, he noticed that they left in separate cargo carriers. In other words, two proteins starting in the same place and going to the same place are taking two different carriers to get there. Subsequently, Emily Stoops in my lab, a wonderful graduate student, did a similar experiment for an apical protein, in this case, a protein called GP135, and found that when it was delivered to the apical plasma membrane, it showed up at the apical surface at the base of the primary cilium and spread out radially and laterally from there, whereas other apical membrane proteins were delivered all over the apical surface of the cell. Alison Zajac showed us beautifully yesterday that the lateral plasma membrane actually is not a uniform target for vesicular delivery, and that some proteins are delivered to its base. What I'm showing you here is that multiple different proteins going to the same place and leaving from the same station take different trains to get there and are targeted to different places. Now, David just set up beautifully that two-dimensional cell culture is not the best place to look at processes that try to recapitulate what happens in a tissue. What about MDCK cells grown in 2 or 3-dimensional culture? Well, David showed very nicely that they can recapitulate certain aspects of tissue or organ formation, but not all of them. So I would submit to you that MDCK cells grown in three-dimensional culture do not resemble a kidney tubule. A kidney tubule is made up of numerous epithelial cell types distributed over the length of the nephron that have morphologies that are commensurate with very distinct functions in these different domains, as Aura showed us. Here in scanning electron microscopy from Keith Porter from about 45 years ago, Dennis Brown from a few years ago, you can see that the proximal tubule cells have beautiful microvilli, deeply infolded basolateral membranes. Here, if we look at more distal segments, you can see again that the basolateral membranes are infolded, here stained with sodium potassium ATPase. And when you look by electron microscopy, you can see that each of these infoldings is endowed with its own personal mitochondria that presumably sits there in order to feed ATP to that sodium potassium ATPase. And we're very interested in trying to understand how those infoldings form, how the mitochondria know to go there. So what's missing when you grow MDCK cells in 3 dimensions? Well, of course, a vasculature is missing, fluid flow is missing. In other words, the physical and biological context for those epithelial cells in the context of an organ. Dave suggested building better dishes for organs. I suggest serving a more interesting food. In this case, since the people working in this on my lab are Italian, we'll call it rana or frog. We're working in an experimental system that is manipulable, but allows us to probe things in a tissue that recapitulates a mammalian nephron. In this case, we're using the pronephric duct of the Xenopus embryo or tadpole. The pronephric duct is essentially a single nephron that recapitulates many of the features of a mammalian kidney tubule, including, as I hope you can see here in this sodium pump staining, basolateral infoldings that are replete with mitochondria. So the question is, can we recapitulate some of the same cell biological experiments that were done in MDCK cells in this Xenopus system in a living nephron in a living organism? So Dave has contributed beautifully to a large literature, as has Keith Mostov and Martin Belmonte, showing that the differentiation of apical and basolateral plasma membranes biochemically is determined in part, at least, by the distribution of inositol phospholipids, PIP45 at the apical membrane, PIP345 at the basolateral membrane, and here using a plextrin homology domain from AKT as a genetically encodable sensor showing the distribution of 345 at the basolateral plasma membrane. When Georgia Skena, a wonderful postdoc in the lab, did that experiment in the Xenopus embryo, interestingly, at least in the proximal nephron, she sees that the AKT is apical rather than basolateral, suggesting the interesting possibility that at least some of the biochemical cues that control the formation of polarized domains may be different in renal epithelial cells in vivo than in MDCKs in culture. How can we intervene in the formation of renal architecture in vivo? So here, again, we're looking at distal tubule cells in the Xenopus pronephric duct nephron, and you can see that the sodium potassium ATPase is distributed in these abundant basolateral infoldings when there's flow. If we use a morpholino approach to block flow, it looks like we can at least partially block the formation of these infoldings, suggesting that flow and the associated shear stress and the metabolic load that it brings may be required for the development of these basolateral infoldings. So that brings us to the challenge questions for Janelia. How can we understand how the environment and functional demands confronted by an epithelial cell in vivo collaborate to determine its structure? How does form come to follow function? And how can we evaluate physiological role and significance of epithelial cell structure? What happens to function if form is perturbed? So we need sensors that report on the mechanical, chemical, and metabolic parameters experienced by epithelial cells. We need non-invasive means for modulating these mechanical, chemical, and metabolic parameters in epithelial cells in vivo. And we need quantitative metrics for describing changes in cell, tissue, organ, morphological properties, as Maha suggested yesterday. To answer the second question, we need what I would call interventional cell biology. That is, tools that can be used to alter cell structure in precise ways in vivo so that their effects on physiologic functions can be determined. And I just want to thank all of the wonderful people in the lab who have been involved in these and other projects, and the great collaborators we've had. And I will stop there. Thank you very much.
Thank you, Michael. That was a really nice talk. Let's move on to the last presenter in this session, Seyham Ibrahim.
Thank you. So can you see my slides?
Yes, we can. Yep.
Okay, great. So first of all, I would like to thank you very much for the opportunity to show you some of my work here today. So my interest is in studying the cell biology of the cytoskeleton and cell membrane in the context of the live mammalian digestive epithelium. Now, we've already heard a lot about the gut, and we know now that the intestinal epithelium is the largest barrier between our internal and external environment. And the whole epithelium is turned over at a remarkable rate of several days. Add to this the complexity brought in by hormonal regulation, innervation, peristalsis, the immune system, blood flow, and the microbiome, and what we have is an exquisitely intricate physiological ecosystem with a very rich cell biology. So can we actually study this cell biology in its native habitat? So first, I would like to show you some ongoing work that we're doing to try to understand the role of the actomyosin cytoskeleton in providing structural integrity to this epithelium, whilst at the same time allowing it to be dynamic. So just to orient you, this is a snapshot zoomed in from the intestine of a mouse that expresses a GFP-tagged myosin 2C. You were introduced to this myosin yesterday by Colby. And as she showed, this is an extremely important myosin, and we found it's really essential for maintaining both cell size and cell shape in intestinal epithelial enterocytes. Furthermore, recent work showed that a pharmacologically activating myosin 2C actually was able to reduce the metastatic load in colon cancer in live mice. So the question we had was, can we visualize this network on this intestinal epithelium in vivo in a live mouse? And the answer to that question is a resounding yes. So here I'm showing you intravital microscopy imaging this myosin 2 network in the intestine of a live mouse. And not only can we visualize it, we can also actually use many of the quantitative cell biology tools that we use in vitro, in vivo. For example, we can use photoablation, laser photoablation, to measure the tension in the network. We can also use FRAP to measure the local turnover of the myosin molecules. We can also track how these myosin complexes move around at the second scale and also over several minutes. And finally, we can use optical flow type algorithms to track myosin velocity across cells, across the whole tissue. And so using these approaches and other data, we were able to come up with an interesting new idea showing how the actomyosin cytoskeleton at the apical surface of these epithelial cells is integrated across the surface of the tissue to impart structural integrity via forming a sort of shell structure. And so how are we able to attain this type of resolution in vivo? I've been working with Bishara Kashar at the NIH, and what we found was that simply by adding magnification to a standard spinning disk confocal microscope, we were able to reduce the effective pinhole, back projected pinhole size, and effectively increase our resolution. And so you can see here that in vitro, this translates to a lateral resolution of about 160 nanometers, which we can actually push to 100 nanometers after deconvolution. And so what I'd like to show you now is how I've used all of these tools and approaches that I've introduced to determine in vivo at the nanoscale how the cytoskeleton drives membrane remodeling in vivo. And for this, I'd like to move to secretion in the gastrointestinal tract. So unlike exocytosis in neurons or clathrin-mediated endocytosis, secretion in certain organs of the GI tract is carried out by secretory vesicles or granules that are an order of magnitude larger. So they're about a micron in diameter. And in order to remodel this large amount of membrane, you need force. So work in the Weigert lab showed that actin and myosin coat fused secretory granules. And these are the machineries that are responsible for generating these forces. And so what I became very interested in is understanding what the architecture of this actin-myosin network is on this micron scaled curved membrane, and what are the dynamics that allow it to generate these forces. And to address these questions, I've been using the pancreas and the salivary gland in the live mouse as a model system. And here I would just like to show you a movie of what this process looks like in vivo. So here is regulated exocytosis in the salivary gland of a live mouse that is expressing a GFP tagged actin marker. And so at this point, what I wanted to do was to use our extended resolution microscopy as well as other super resolution microscopies to interrogate the architecture of actin and myosin on the fused granule. And what we found, which was very exciting, was that myosin 2A forms this three-dimensional cage lattice-like organization around the membrane that had not previously been described. And this was very exciting to us. And we saw a similar structure for actin. And so the question was, well, how are these cages moving to generate forces? And so to address this, we once again went intravital. And here is a movie showing the myosin 2 dynamics during exocytosis in the salivary gland of the mouse. And you can see the blood flow very nicely here. And of course, the benefit of imaging this at a high resolution is that you can actually zoom in on individual granules, and then you can visualize and quantify and analyze how this cage is assembled, how it changes over time, and how it eventually disassembles. And by measuring things like the fluorescence intensity changes over time, fluorescence density, and the diameter, we were able to infer that this myosin 2A cage was actually contractile. We were also able to do FRAP in vivo of these individual granules, and our FRAP analysis basically validated the contractility of the network. And so what I just wanted to highlight here is using a cell biology approach in vivo, we were able to reveal a completely new architecture of the actin-myosin cytoskeleton, and also dissect the mechanisms of how they work together to generate forces to remodel membranes in vivo during secretion. Moving forward, I've become increasingly interested in septins that are the newest, let's say, component of the cytoskeleton, and that intimately interact with both actin and myosin, as well as curved membranes. So I was very excited to see that septins are also present on the secretory granule and are likely new components of this membrane remodeling machinery. Septins are also extremely understudied in the intestine, and I found that they actually interact intimately with actin-myosin at the terminal web, and also form these long-term, long-range scaffolds around specialized epithelial cells in vivo. And I've now generated mouse models, both knock-in mouse and knockout mice, to examine septin cell biology in vivo. So what are the challenges? There are many challenges to doing this type of in vivo cell biology imaging at a high resolution. Probably the biggest challenge is motion, because these animals are living, breathing with a heartbeat and a blood flow. There's a lot of motion involved. And so faster imaging and or post-processing approaches for motion correction would be amazing. Similarly, because we're imaging in tissues that are very heterogeneous, causing wavefront distortions, approaches that can correct for these would be very useful. So most of the data, or all of the data that I showed you today, was acquired within sort of 100 micrometers of the surface of the tissue. And of course, if we want to go deeper in, we're going to lose that resolution. So ways to break this penetration resolution trade-off would be amazing. And like others before me have said, genetically coded functionalized fluorescent probes to relate the cell biology that we're seeing to physiology, such as markers for metabolism and signaling, would be extremely useful. I think Janelia is in a unique position to actually integrate all of these synergistically and hopefully make it accessible to the community as a whole. And with that, I'd just like to thank my lab, the Weigert Lab, my mentor, and the Kashar Lab, with whom we collaborate extensively. Thank you very much.
Thank you so much, Sam, and thank you to all of the speakers in this session. And now we will turn it over to Denise Montel and Wallace Marshall for the discussion period.
Okay, I think Wallace is going to start off by leading the Q&A.
Okay, yeah, so we're going to start with some questions that came up from the Q&A box, of which so far there's only one, so please, oh, there we go. So first we have a question for Dave from Agata Naiga, which is, if you have these different sort of structure types coexisting in the culture, are they communicating with each other?
You broke up a little
For example,
bit.
you know,
read
do
the
they
message,
secrete
so
factors that
the
the others
okay.
respond to?
Yeah, so this is a really great question. We've been doing this in prostate cancer, and we have done some of the things that we're proposing Janelia could do, but we're a little mom and pop shop type lab. We're quite small, so we've done it in a rudimentary way. But we have some definite answers, which is that we can see up to sometimes 16 different phenotypes occurring in parallel. And when we isolate some of these phenotypes, they're stable. And they can be recapitulated so that they all do it at the same time. But what's very interesting is that when you have them apart or together, they do these phenotypes independent. So it seems to be that some of these are cell autonomous, whereas others that we've picked and tried to further characterize really generate a few different phenotypes, and they seem to be doing it in close proximity. So this suggests that there's a combination of plasticity as well as non-autonomous effects. And so I think that we get both completely independent phenotypes as well as phenotypes that are dependent on each other and also are interchangeable.
Does it matter how far away? I mean, do you know what the communication is mediated by?
That's a great question, but it's very hard to answer. If you would need a way to determine how far apart you place them in a regimented way. But by doing that, you're affecting their ability to interact. So I don't really know. It's the case of, you know, you perturb to observe too much. In terms of the phenotypes that are maybe transmitting to each other, we don't know some of the signaling molecules. We've mostly focused, for reasons that you'll probably immediately understand, on the phenotypes that are a lot more cell autonomous and locked. They're a little bit easier to understand. But you know, what I would say is that heterogeneity is the rule, not the exception, and that everything we've profiled, patient-derived organoids, mouse model organoids, cell lines, everything is heterogeneous if you have the tools to understand distinct states that occur repeatedly. But to me, the most shocking thing, maybe because I'm naive, it's shocking, but makes sense in the end, which is that if you look in most of the samples we look at, the ratio of different phenotypes that are occurring in parallel is remarkably consistent across experiments done years apart. So it seems like there is a ratio of the different phenotypes that can occur, and this is largely set. I had initially thought 3D would just be extremely noisy, and it is, but it's quite remarkable. And so I think when we think of a cell line or a sample, what we really have are cells somewhere along a plasticity continuum. And we have to be careful when we talk about what is the phenotype, because depending on how we do it, it's probably the phenotypes in parallel. That gets a bit scary when we pick clones for CRISPR, and it gets a bit scary when we think about transplanting, because which is the phenotype that's happening?
But it's cool because it seems like it's inherently then four-dimensional, even the idea of what the phenotype is. All right, so we have another question in the Q&A from Wyatt Korf, which was inspired by Michael's talk, but really it's for everyone, which is, how much is known about, this is a big question, how much is known about the role of physical forces like tension and flow in development? And I think the correlate really is, to what extent should that be incorporated into any kind of attempt to recapitulate development in a dish or other kind of system?
So the short answer is not a lot and yes. I mean, there's a huge amount to be learned about the extent to which physical forces, dynamic events like flow, like pulsatile changes in delivery of nutrients or oxygen, et cetera, plays a role in determining what cells decide in terms of the shapes and structures that they will assume. Modeling that in vitro is extremely hard. I mean, one of the things that has been alluded to briefly in the context of organoids is that so far kidney organoids are sort of disappointing. You can get differentiation into different structures like glomerular cells, proximal tubule cells, but they are pretty much trapped at a very immature stage. And almost certainly that has to do with a lack of vasculature and a lack of flow. So I think that developing either bioengineered systems, as has been talked about in the previous set of talks, where you can superimpose flow and rigorously control delivery of soluble signals, et cetera, and or working in the context of an organism that makes some of these high time resolution processes accessible, as Siham beautifully did, or as perhaps the frog can provide. I think those are going to be ways to try and work from both ends to address that problem and meet in the middle.
Yeah, and I think what I would add to that is that, with the exception of skin, epithelial organs, their point is to allow fluid to flow. They want to build a lumen. They want to transport various different things. So part of their entire function is to create those gradients, which will, of course, incur force. And all of the models in 3D that are spheroids, luminoids, whatever you want to do, are terrible about that. The transporters are in the wrong way. They're trying to pump things out through the basolateral membrane. So I think these models tell us something about the design principles, but they're halted at a particular point. And I think what Jim showed earlier today with the Lutov's labs microfluidic dikes are one way towards that. But I think the throughput of those are also extremely low. So I think there are many challenges ahead.
All right, then we have another question in Q&A, this time for Siham, which is, what signaling mechanism would regulate the myosin contraction and relaxation during secretion, and how would that work?
So as far as we know, the myosin is activated by phosphorylation, so by like a myosin light chain kinase. And in terms of how we would image that in vivo, by it being able to image markers of phosphorylation or a phosphorylated version of the myosin 2.
But would that be like use like nanobodies or something? I mean,
I'm not entirely sure. That could be one option.
Well, I have my own follow-up to that, which is how do you think it's sort of, is it local to the different, I mean, within one cell, will you have multiple of these vesicles at different phases of their contractility cycle?
Yeah, so the whole thing is happening in vivo. And so the way it happens is you have your, at the apical surface of these secretory cells, you have these canaliculi, which are sort of tubule-like, and they're bound by membrane, and they're covered with actomyosin. And so on beta adrenergic stimulus, the cell receives a stimulus and causes this actin to move away and allows these secretory granules to fuse with that membrane. Now, because the granule is a micron in diameter, and these tubules are only sort of 0.4 microns in diameter, that results in a difference in membrane tension So the membrane tension around the canaliculus is much higher than the membrane tension around the vesicle, which would cause the membrane to flow outwards towards the vesicle, which is why the cell needs to build this sort of cytoskeletal cage and push the membrane inwards. And so once the granule reaches a diameter that is equivalent or close to the diameter of the canaliculus, so around 0.5 micron, then this cage disassembles, and then the rest of the integration can happen spontaneously.
Maybe we'll move on to a couple of questions in the chat from our panelists. Probs, you had the first question up there. Do you want to?
Oh yeah, so actually Michael answered a little bit, but I can expand a little bit on that. So this was about the trafficking of like the different trafficking modes that is followed by these proteins in the epithelial cells. And like Michael mentioned, that it seems that the environment of the cells, like when it's in vivo compared to when it's in vitro, it can make a difference. So I was thinking, like, when you think of trafficking, like there's always multiple trafficking pathways available for proteins, right? Even if you take the most simple cells. So one way to think about it, and I think most cell biologists think of it that way, is that it's like a partitioning mechanism. Like if a protein likes one pathway, preferably it takes that pathway, but then if something else happens, there's some kind of a saturation going on, it can also revert to the other pathway. But the intrinsic, basically the mechanism of the pathway in terms of the players, they are still maintained. So my question is that what's happening in vivo, or is it that now you have a completely different players which are coming into the picture, and you have a completely different trafficking pathway which is being generated?
So I think that's a great question. And I think it reflects in part, to some extent, that the way we look at trafficking pathways is perhaps overly binary. So we, at least in terms of epithelia, we tend to think of proteins are destined for the apical membrane or the basolateral membrane. But really, I think proteins are divided, as Probs was saying, into multiple different sorting pathways that are directed to where a specific epithelium needs them. So for an extreme example, the sodium potassium ATPase in the kidney, as I showed you, is beautifully basolateral. In the choroid plexus, in neuroectoderm-derived epithelium, it's apical. But other proteins that are basolateral in the kidney are still basolateral in the choroid plexus. In other words, the sodium pump doesn't carry an apical or a basolateral signal. It carries a signal that says, I am the sodium pump, put me where you need me. And similarly, other proteins do the same so that epithelial cells can be maximally plastic and determine their physiologic function by figuring out how to distribute these different transport proteins. I think that what differentiates different epithelial cells is what machinery they possess that interprets those signals and decides where they're going to go. In other words, how they choose to read them and how they effectualize the targeting that is embedded within that signal.
Yeah, and I think also some really fantastic work has been done by Fernando Martin Belmonte using zebrafish along with Michel Bagnat, and they showed that different regions along the intestine of the zebrafish, the lumen of the zebrafish gut, express different trafficking machinery. And that this is really deterministic for what molecules are taken up in what part of the intestine. And so I think the idea that trafficking pathways are just set and that molecules will choose to go in one pathway or another is too simplistic. I think the transcription factors that control differentiation of different types of epithelia do that by invoking the machinery that's required to process the molecules that would normally function within those epithelia. So I think in terms of differentiation of different epithelia, it's really turning on the machinery to handle the functions that that specific epithelia uses. And part of that is the trafficking pathways. And they can be very different in neighboring segments of the same tissue that we think of. And this is really determining how they function and interact with the contents of the lumen, as one example.
And expanding on that, is it also possible that the cargo itself can sometimes actually turn on a particular machinery for trafficking? So that like you have an expression, a certain kind of epithelial cell expressing a certain kind of protein at a much higher expression level. And that might actually have a feedback mechanism of what kind of trafficking machineries are actually in operation in that cell.
Yeah, a hormone receptor is a very good example of that.
So I think that
we have a couple of questions for Sihan, one in the Q&A and one in the chat. So maybe we can just pull those together, which is they want some of Daniel and Francis Ashcroft would like to hear more about the intravital imaging of the pancreas. And could you say something more about that?
Sure. So it is tricky to set it up. I mean, you just wanted me to talk through how it's basically.
Well, yeah, they wanted to know if you looked at the role of.
It's basically you make a little. It's a surgical incision in an anesthetized mouse to expose the pancreas, and it's as simple as then just placing that tissue on an objective. Right now, we do a lot of stabilization using sort of tape and just things that you find in the lab. It's nothing too high tech at this point, but we are still able to image dynamics at very high resolution, even with this sort of rudimentary approach.
And do you see the same kind of distribution of septin and myosin function
in pancreas?
In the
yes. Yeah. Mm-hmm.
Mm-hmm.
Can you see the islets?
So I haven't gone that much in depth. So currently for this project, we were just really focusing on the exocrine pancreas and this specific regulated exocytosis. So the short answer is I don't know yet.
So we have a question from Tamala Kui for Ziham about myosin. Tama, are you there? Do you want to?
Yeah, I was curious about the apparent change in the stoichiometry of the myosin 2 minifilaments as the structure shrinks. At least the model figure that you had sort of suggested that. Is it something that you think is taking place, or?
So that is a very good question. At the resolution that we were able to image, it is not possible to really determine the stoichiometry. So the inferences that we made were based on fluorescence intensity changes. So if you look at the intravital movies, there was a point at which your fluorescence intensity stops increasing. So you don't have any additional myosin filaments being added to the granule surface. And yet the fluorescence density goes up as the granule is shrinking. And so from that, we inferred that the filaments that were already on there are getting more tightly packed. And so that's likely.
So you rather think of sort of stacking minifilaments, and that makes them brighter.
Yeah,
brighter.
Exactly.
The other issue then is that of length. So I don't know. In your model, you sort of put minifilaments drawn onto a lattice. In your movie, it was clear that actually the nodes, the vertices of this lattice were actually getting closer.
Yeah, exactly.
So,
and this is something.
Do you think that the minifilaments? So what's in the nodes? What's in the edges? Do you think you have consecutive minifilaments that are slide onto one another, or are they actually changing their organization altogether?
So the nodes represent the head domains of the myosin. So the GFP tag in this case was on the head domain. So when the nodes are getting closer together, it suggests that the bipolar filament is actually shrinking and contracting.
If that is true, I think is a new observation. I don't think it's never been made before. It's very interesting.
So it is very exciting, and it would definitely be very important to further investigate it. I think we're just at the tip of the iceberg for now.
So we have some hands up. Should we?
I was noticing we had Q&A.
Oh yeah, we have both. Yeah. So
I'm not seeing the hands up. Do you want to go? Why don't you take the hands up?
So I'll do some hands. So I think the first one to go up was Patrick Chitwood.
Oh, yes. So I guess I'm kind of interested in this, this idea of recapitulating what happens in a tissue and in a dish. And I guess when considering both the talks that have happened previously today and then also these talks, there's so many factors, it almost seems like an impossible task. That might be naive because I'm not quite familiar. But I'm also wondering if it's even necessary to fully recapitulate, or if in a dish you really just want to recapitulate one or two components, investigate those thoroughly, use what you learn there to really inform targeted questions that you can then go in vivo with. And so what could be really nice is setting up a system in which you have two investigators working in parallel, one working from a bottom-up approach, one working from a more top-down approach, and sort of collectively working together to try and get to the answer. Or do we think we can really recapitulate something in a dish, or these other models described?
But I think you can definitely recapitulate things in dishes. I think it depends on what those things are. I think, as has been alluded to, some systems are more amenable to growth as organoids or in dishes than others. The intestine, for example, seems to do a great job. The kidney does sort of a miserable job. And I agree completely that working in both directions makes great sense, and that using tractable genetic models that allow in vitro perturbation of cell biologic processes, as well as simplified systems in a dish, allows you to try and manipulate things and to sort of assess complexity and meet in the middle, to try and figure out which components are important for creating a structure, allowing you to either recapitulate it in vitro or perturb it in vivo.
Yeah, and I think it really depends on a matter of resource. I mean, do you want to be a lab that stretches yourself across many different things from in vitro to in vivo, or do you want to be part of a network? I mean, here we do what you have suggested. We do the in vitro work, and we transplant what we do in vitro back and graft it back in vivo in intestinal work. And that works supremely well to go in vivo to in vitro, but you need the resources and the shared collective interest to do that. And that's not always achievable.
So Sylvie had a hand up.
Yes, so my question is for Siham. Very, very beautiful talk.
Thank you.
I was impressed by the resolution you could achieve in your in vivo model. And also so how did you achieve such a resolution? Can you let us know? And you also, I have a couple of, and you have also mentioned that you had, as usual, in vivo imaging, you will have breathing and heart beating. So how did you actually get rid of this movement in your imaging? It was really nice.
Sure. So in terms of the resolution, it is a bit of a Columbus egg situation. So I think it's common knowledge that if you reduce the pinhole size beyond 1 area unit, you do get an improvement in resolution. And that is pretty much exactly what we did. So we just added some additional magnification between this microscope and the spinning disc, and the images looked better. And then when we got these sort of calibration tools, the commercial ones, we were able to validate that indeed we had an improvement in resolution. So I think the best thing about this approach is it's so simple. It's so accessible. Like I said, there was nothing really specialized about it. The Nikon spinning disc that we have already comes with a 1.5x tube lens, and we just added an additional 2x lens to that. In terms of the motion, that's a great question. So a lot of the stabilization, as I said, we do using like pipette tips and tape and cotton swabs just to kind of stabilize the animal or move the moving parts away from the objective. And then I actually just used the Nikon Element software that's pretty decent at doing alignments and corrections, surprisingly. So very simple answer to both questions.
And what's the time course? Yeah, I know there were movies with time scale, but I missed that. So what's the time course for a granule to form and fuse out or detach?
So what happens is it fuses, and then it takes about a minute for the whole thing for the whole membrane to integrate. And so the movies that I acquire are in the range of 10 minutes for salivary gland imaging, and for the intestine, we've done close to sort of an hour even. And the intestine is more challenging to stabilize and correct for.
So you anesthetize the animals with what?
So we use ketamine, xylazine.
Yeah.
Okay,
Because
I think we're going to go to Manu quickly, and then we'll go to one of our audience members, Nick Cronin, for their questions, and then we'll come back to GT. So Manu.
I'll keep this very quick, but I was curious, many of you discussed the lack of fluid flow in organoids and the lack of tools associated with this. And I wonder, as a community, it's sometimes helpful for tool makers if there is a very prescribed regime of fluid flows that would match a large number of organoids. Actually putting numbers to this is extremely crucial, because when you think about the breadth of biological fluid flows, it's quite extreme. And sort of an idea that I've been thinking about, and I just want to toss this here, is sometimes we often think of driving flows from the outside, which is challenging because you have to build an interface. But there are new techniques now to be thinking about internally generated flows, and especially active flows that are generated in closeds. So it's one way of decoupling the idea that flow can be there even without an external transport mechanism. So I was just curious if somebody's actually put together a needs list. That would be really fun to see.
So I'm not sure that I can say that a needs list has been put together, but I will certainly agree that the magnitude of the flow is a critical variable, and one that has to be directly tied to the system that you're looking in. So for example, the flow rate and shear force in a kidney tubule are very different than they are in an intestine. And there's pretty good evidence that the magnitude of that flow can make real differences in the signaling properties of those cells. Ken Spring, who retired from NIH, did beautiful work on measuring flow in tubules, and in fact, he and Hella Pretorius were the first to show that the bending of the primary cilium under biological magnitude shear stress sent signals that were interpreted in the cell and that sent messages to downstream cells. So I think it's critically important to match the flow properties to the tissue from which the cells are derived.
All right, so maybe we'll take, there's a question from the Q&A from Nick Cronin, which is, if you change nutrient availability on one side of a monolayer or organoid, will that then cause a switch in apical, basolateral apical trafficking to, I guess, to better access the nutrients that are there?
So I'm not aware of anyone showing a switch in polarity. There's an old literature actually from Kai Siemens showing that if you grow MDCK cells, for example, on filters instead of on plastic, they become more polarized. And the theory here is that epithelial cells put most of their nutrient transporters on the basolateral surface because they're used to eating from the bloodstream or the extracellular fluid compartment. If you grow them on plastic, then they can't do that anymore. And in fact, the ratio of distributions of proteins is less polarized when cells are grown on plastic than on filters. It would be great to see if there were a metabolic switch that controlled distribution of a specific protein, but I'm not aware of it yet.
I can weigh in on that. When we grow organoids in vitro and then compare glucose transporters to organoids that we grow in vivo that become vascularized, there is a switch in glucose transporters in terms of localizations. I honestly can't remember whether it was GLUT4 to GLUT2 or GLUT1, but there was a switch based on, in vitro versus in vivo growth. And then those can be, and then you can affect transporter activity with these tissues by either adding glucose or other nutrients after you reharvest them. But yeah, they do move around depending on whether they're grown in culture or grown in a more physiologic setting.
Yeah. Thanks, Wallace and Denise. Yeah. So I was just noticing from the presentations that looking at the diversity and the and the plasticity of the different cellular trafficking mechanisms that we are seeing, as well as organelle positioning in sort of very closely related cell types within an organ or within millimeters or no, sort of micrometers in a tissue. I mean, is an incredibly interesting problem, because we are seeing this diversification of in some ways cellular plans, which would be, I think, something to take heed of and see, if we can then combine this with this intravital imaging that we heard from Sehan. We, I think, are around to something very, I think, important about how the architecture of a tissue can be constructed from these unitary processes that we know about from studying cells in a unpalatable 2D dish. So I just thought I'd throw that thought out to say that it's important for us to look at these various plans that can emerge and see what sense one can make of them. I mean, there's so much to learn. I mean, what are the signals involved in creating these sorts of cellular plans? What are the mechanics that influence these processes? What sort of short and long range communication that helps to set up these patterns? And of course, if these patterns are stereotypic, then what regulates the whole system? So I just thought I'd make that comment. Yeah.
Well, there's also a comment from John Hammer saying that intravital is perhaps the most physiological context to do imaging. Can Janelia do a ton of knock-ins, or at least some epithelia-focused ones in the mouse to support this approach? And maybe, because we're getting close to the end here, I would like to throw out to our three speakers, we've heard a lot of people write very long wish lists for a sensor for every metabolite and every signaling pathway and every forces and also ways of manipulating all of those different things. If you can only have one, which one would you choose? Siham, why don't you go first? And then we'll go to Dave and then Mike.
I think sensors for metabolism and would be very important across all the different questions that we've heard over the last 2 days. So I think if I had to pick one, that would be the one.
Be for metabolism, but so what metabolite?
What metabolite? Do I have to pick one?
No, you can think about that. Everybody can think about that. Dave, if you could only have one of the things you've wished for, what would it be?
Software solutions to help heterogeneity analysis.
Mike?
I would say shear stress. That's the force that, at least this week, I'm most interested in.
All right, thank you guys so much. This was a great session, really wonderful talks and really enlightening discussion. Thank you, Denise and Wallace, for leading it, and to all of the speakers. And with that, I would like to turn it over to Jennifer Lippincott-Schwartz to introduce our last session of the day.
All right. So we're going to go into the last session, session 8. Really been an amazing 2 days. This session is entitled Single Cell 40CP in Renal and Intestinal Epithelial in Vivo. Dennis Brown is going to lead off. He's a professor at Harvard University. Then Knouse Peti-Peterdi from University of Southern California will follow. Then we have Catherine Schrankel from the Scripps Institute of Oceanography. And that will be followed by our discussion and Q&A, which will be led by Sylvie Breton, associated with Mass General at Harvard, and Prabh Gupta from Janelia. So why don't we get started? Dennis, do you want to share your slides with us?
Okay. Got them?
Yes, very good.
Okay, so thanks for this opportunity. And I think one of the most exciting things, as in my capacity as the Chief Science Officer for the APS, is to hear the word physiology said so much over the last 2 days. So let's just keep it up. I'm going to talk about our work on the kidney, but not so much the latest work that we've done, but just put it in the context of the types of things we've been asked to talk about. Let's see. So the kidney was set up by Aura and by Michael, and it's an extremely complex organ, as you know. Here, this is an India ink-injected kidney with all of the little filtration units, the glomeruli in black. But each human kidney contains about 1 million, on average, nephrons, which all work together in a coordinated way to maintain body homeostasis. And this is the nephron, as you've seen. There are many, many different types of epithelia. I don't want to get into we have more cells than you have in your organ debate, but there are about 30 or so different cell types in the kidney, many of which are epithelial cells that do various jobs. And the way that we normally break these down and look at them in the intact kidney is by morphology, immunocytochemistry, such as here, where we've used an opal technique to do multiplexed imaging of in 6 colors. And what we can see here is that when we merge them together, that these make a beautiful mosaic, and the most colorful ones are the collecting ducts and the distal tubules. And it doesn't really matter what these transporters are, just understand that it's extremely complex. They're all over the cells. Different cells express different transporters in different membrane domains. And so the question is, how can we go about dissecting this and this function in the kidney? And so the challenges that we're facing are really high resolution functional imaging in vivo. We can do the static imaging, now we need functional imaging. And you're going to hear some more about that from, I think, from Janos and Kat in the next 2 talks. So we need physiological responses. We know that the kidney responds to cues, to metabolic cues, to hormonal cues, and changes epithelial function. We know that there are markers of transporting signaling processes that we can follow, and we'd also like to follow cell fate mapping during development, during repair, and actually during normal kidney function. The tools and reagents that we need are expression systems in the membrane and cytosol that allow us to do this. Reagent penetration and access are a bit of a problem in the kidney because of the glomerular filtration barrier, but nanobodies are that we've heard a lot about today or yesterday. Nanobodies might help us to conquer this, at least at the apical pole, by passing the glomerular filtration barrier. And then we need the microscope and tissue preparation technology that will allow us to do this, and which Janelia is so good at. So looking only at the collecting duct, this is a longitudinal view of a collecting duct. The green aquaporin 2 and the red aquaporin 4 on the opposite poles of principal cells, and the vacuolar proton pumping ATPase at the apical pole of the here of the intercalated cells allow proton secretion, allow water reabsorption, but also these cells are involved in potassium transport and sodium transport, as well as many other things. And how can we chop this up and dissect it and look at the function? So first of all, before I want to talk about that, let's look at cell shape changes. I want to go to the epididymis, which is a favorite tissue of Sylvie Breton, one of the panelists here, and also the kidney. So Sylvie has used, over the last few years, the epididymis as a kind of a model system for the kidney. And what you can see is that in this particular cell, which was thought to be a basal cell, we now know that this extends a huge process all the way up through the epithelium and pokes through the tight junction here. And we know that it samples the luminal environment and prepares the tubular lumen for the arrival of sperm. So what we were able to do, because the epididymis is already outside the bulk of the body in the scrotal sac, it's possible to use 2-photon microscopy to follow these cells expressing a red keratin. And what we found is that these cells change the length and the extent of their process in a process that we call periodic axial motility, or PAM. And we can see that these oscillate, and on the right you can see that the basal cells are not really, some of them are basal, but some of them extend these processes that go up through the lumen between the tight junction in this sinusoidal oscillatory way. And just for the trainees on the call, if you want to get a paper into a Nature journal, you have to invent the name of a process. And we call this PAM. Initially it was called SAM, but we changed the sex. We said it was sinusoidal axial motility, but somebody pointed out that it wasn't really sinusoidal, so we call it PAM now. And in this particular case, they're angiotensin II sensing cells. So proton secreting cells in the kidney called intercalated cells also change their shape during activation. Here's an inactivated cell with lots of vesicles containing proton pumps. And when the body gets too acidic, they get activated. They secrete protons by incorporating pumps into a very, very complex apical membrane. Those of you with a microscope in your glasses can actually see the gold particles on the immunogold labeling here in the vesicles. So for those of you who missed this publication when we published it last century, these vesicles are extremely well endowed with proton pumps, and we can look at these by rapid freeze deep etch proceedings, and this is what they look like. Each of these balls here is about a 20-25 nanometer proton pump, and these are the ones that flick back and forth between the apical membrane and these cells. By helium ion microscopy, this is what the cell looks like when it's been activated. This, by the way, is a cilium, a single central cilium on an adjacent cell, and we can go even higher and look at these beautiful infolding on these cells. So we'd like to see more of this in the kidney and in other organs as well. So functional changes in transport. Well, in the epididymis again, we can put a self-referencing microelectrode adjacent to the epithelium, which we can cut open. And again, this is work done with Sylvia Breton. And we can add angiotensin II, which stimulates proton secretion. And this is inhibited by concanamycin A, showing that it's VATPase dependent. But this is really difficult, if not impossible, to do in the kidney. So what we'd like to see is to express a marker on the apical pole of intercalated cells in the kidney. And I've used pH here as an indicator, but we can look at sodium, potassium, phosphate. Any marker that can detect any activity of these cells can be expressed on the apical pole. The green marker here is actually the VATPase. When we stimulate these cells, this marker may turn a different color. It may change intensity, and we would have a real-time accessibility to see changes and differences at the apical and, in fact, the basolateral pole of these cells in real time. So that's one of our asks. So cytoplasmic signaling. Well, we've known for many, many years that in the kidney and in the other organs, we can express in a cell-specific manner green fluorescent proteins, in fact, a variety of fluorescent proteins. This case, this is EGFP in intercalated cells. And you can see at higher magnification that these green cells express the GFP, and it would be easy to envisage that this could be a marker for a metabolite, for example, for calcium, as you'll see from Janos. And this is one of our asks again, that we can make an array of markers in the kidney expressed in a cell-specific manner that would be applicable to other organs and would be accessible to in vivo microscopy. So membrane protein expression. A lot of cells in the kidney and other organs change their function by expressing membrane proteins, by shuttling proteins back and forth from the cytoplasm to the membrane and back again. One of the ones we've looked at over the years is aquaporin 2. We know that the kidney is involved in concentration. It responds to ADH, the antidiuretic hormone, by moving aquaporin 2, in this case on these little vesicles here inside a collecting duct. This is the lumen, this is the collecting duct. This is a Brattleboro rat kidney, which does not express ADH. It doesn't have ADH. You inject ADH into these animals, and 10 minutes later, this collecting duct, they look like this, that with the aquaporin 2 firmly embedded in the apical membrane, which allows this cell to reabsorb water back into the body. So this shuttling process is something we'd like to see in real time in tissues in the body. We're looking at ways of drugging this process to inhibit it in hypertension, for example, to stop water being reabsorbed, and to stimulate it in cases of diabetes nephrogenic insipidus, where you produce too much urine. We'd like to reduce the amount on the membrane. So this is a transverse section of a cortical collecting duct, and in green is the vacuolar ATPase, and these are proton pumping cells. But look at this one. This one has basolateral ATPase in addition to a little bit of apical. This is called a B cell, a B intercalated cell. And there's been a big controversy over the years as to whether this cell can actually transform into this type of cell. Michael Caplan alluded to this earlier on. Can this protein change polarity? And what we know is that in the B cell, it probably can. But what we don't know is whether this alpha cell can change into a B cell. And so what we'd like is to use a fluorescent protein probe or nanobodies, etc., that we can deliver to the lumen, that we can express in the cells already. And we would like to see in real time whether one cell can change polarity by moving the VATPAs from apical to basolateral. In other words, we'd like to follow this trafficking process in real time in the kidney. The green here is aquaporin 2, and we'd like to follow trafficking of that as well, again, using nanobodies. So again, we have these challenges in epithelial biology. We'd like to do high resolution imaging in vivo. We'd like to look at physiological responses to various cues in the body, acid-base cues, volume cues, hormonal cues, etc. We'd like to look at the dysregulation of the transport and signaling process in real time in disease states. And we'd like to look at cell fate mapping to look at kidney construction during repair and during renal damage. For that, we need expression systems that express these proteins in the membrane, in the cytosol. For example, apical membrane proteins could be attached to a GPI-linked protein, or apical membrane markers to a GPI-linked protein to express it in the apical membrane. Reagent penetration could be solved by, for example, the preparation of nanobodies against every cell, every protein in the genome, as we just heard about yesterday. And we'd like microscope and tissue preparation technology to be improved. And I think maybe Janos is going to talk about 3-photon microscopy that can improve the depth. But all of these things together are applicable to the kidney, but also to many other organ systems. And then finally, I'd like to leave you with a picture that my daughter-in-law sent me for my birthday last year, which is a couple of cute little kidneys. Stop looking at me, I'm trying to concentrate. So that's it, thank you very much.
Okay, we cannot hear you, Jennifer.
Jennifer, you're muted.
Thank you very much, Dennis, for that really wonderful talk. We'll now move to Janos.
Hi, everyone. Hope you can hear me well, and you can see the slide projecting already, hopefully. I really appreciate the opportunity to contribute to this really excellent workshop. I really enjoy all the talks. So here, I'd like to continue with Dennis's theme and provide our contribution focusing on in vivo single cell for the cellular physiology, and use the nephron central command, a specific cell type within the kidney, as an example.
Janos, can you put your presentation in? Yeah, there you go, full screen.
Sorry, I thought I already did. Sorry about that. So as a general introduction of our lab, we, with a physiological foundation, specifically in pathophysiology and regulatory mechanisms of body fluid and electrolyte homeostasis, our team approaches the mechanisms of endogenous tissue repair, which we've heard in other talks focusing on other organs with the same approach. We would like to understand and augment endogenous tissue repair in a way to develop new therapeutic, regenerative therapeutic approaches for chronic kidney disease. But I wanted to emphasize that we do this with the physiological foundation. So for the cell type of our interest, and as an example, the model system that we use in the lab, we focus on a very mysterious but beautiful cell type, which is called the macula densa cell, which is a group of about 25 cells in a tightly packed location at the vascular pole, the glomerular entrance of the blood vessels that bring in blood for the filtration. This cell plaque is illuminated in green color, so you can better appreciate the strategic localization and perhaps reflecting on the function of these peculiar and mysterious cell types. Importantly, these cells, although being a totally beautiful polarized epithelium, they show significant signs of neuronal differentiation, so we can call them neuroepithelium, and they really work and function as the nephron central command, namely to regulate the amount of blood that flows into the kidney filter for filtration, the amount of renin that is released from the adjacent renin cell type to drive the activity of the systemic renin-angiotensin system, and so forth. Importantly, macular densa cells can detect alterations. They are excellent sensor cells. They can detect anything that's in the extracellular environment. if it's being the ionic composition of the tubular fluid at this point of in the early distal part of the nephron, namely sodium chloride concentration in the tubular fluid, but also mechanical stress. MD cells have the longest primary cilium in the entire nephron. Also, they have excellent metabolic receptors, G protein coupled receptors of many metabolic pathways, systemic hormones that have been recently identified in these cells, really working to detect local and systemic environmental cues. So they represent a cell type which is playing a very important role in interoception to detect the intercellular milieu and provide physiological responses to regulate organ function and alterations in the whole organism. So I wanted to list some of the concepts and the approaches that I feel would be important contributions to this workshop today. Namely, I cannot emphasize enough, and this is a good audience to emphasize this, physiology is really the key to understand kidney tissue repair and in general, organ-specific physiological stimulation to any type of tissue repair and the role of physiological adaptive mechanisms in this response. And more specifically, we focus on chief cells in organs that respond to organ-specific physiological stimulation that drive endogenous tissue repair. For example, in our own case, this is the loss of body fluid and salt in the kidney, but similarly is fasting in the GI tract and so forth. Another important concept is that it's the response to the environmental cues, namely physiology, that drives complex genetic programs of tissue repair and so forth, and not really genetic programs have the priority. We do need the physiological understanding in this to interpret genetic programs. And needless to say, for this workshop is that we need to reverse this general reductionism that we've seen over the years in research to study cell, organ, and organism function back in the in vivo environment. And again, consistent with the theme of this workshop and my talk specifically, emphasized throughout the role and the need for single cell resolution needed to understand physiology due to a variety of facts, including cell heterogeneity within the tissue, hierarchy within the tissue, and all the dynamic changes in cell biology, physiology, including metabolism that we've heard so much already in this workshop. So all of this leads us to the need, and also I'll show you some examples how we use in vivo single cell for the cellular physiology, or using the term SC4-DCP. In our approach, such as you've heard from other organs, pancreas and liver and skin so much in this workshop already, but we apply this for the kidney ultimately to use serial intravital multiphoton microscopy. And the emphasis is being on the serial feature of this. So we look inside the intact organ, the kidney, and to observe the function and the alterations in the function of the same single cells, vascular, tubular, and structures in the exact same kidney and the mouse over time. An example is the use of these abdominal imaging windows that many of us have been using, widely used for a variety of abdominal organs. In this case, we apply this for the kidney, implant dorsally a metal ring with a cover glass glued on top, and you can see the kidney being inside in direct contact with this. And so we can put the exact same animal with very brief isoflurane anesthesia on the microscope stage daily, if you wish, to find the exact same tissue areas, in fact, create a map of the tissue and do this over and over again and see how the cell distribution within the same tissue volume changes over time. To provide you with examples how we apply this in vivo SC4-DCP in our work, again, we combine intravital MPM with genetic mouse models to track changes in single cell physiology over time and in vivo, both on the structural and functional level. So one example I like to use this on the structural side is to use this MD-GFP mouse model that we developed recently with a macular cell specific expression of GFP, specifically in the cell membrane, targeting with the MTMG approach. And importantly, we use, this is a Krelox-based system, inducible, but importantly, we have to apply for a mosaic type of expression, only a very low dose partial induction with tamoxifen. So on two panels show on the left-hand side the difference, what we have with total for full induction of the GFP expression, showing all the cells. You can clearly see individual cells in the entire MD cell plaque. You can count the 25 cells in this image. Importantly, there's not much detail on the cell membrane. you cannot really see subdomains of the membrane with this full induction. You need single cell illumination here. And partial induction allows us to do this. An example is at the bottom of the image. In this case, if you give only partial induction, you have the ability to visualize only 1 or 2 cells in this case, in which you are now able to see these beautiful, so far unknown axon-like, very long processes of the lateral and basolateral membrane that go very, very far, 50, 60 micron away from the somata of these cells to form junctions with neighboring cells. Again, emphasize the role of single cell resolution. On the functional side, I show you, for example, a very general and ubiquitously used reporter, the GCAMS, as we've heard before, specifically SALSA-6F, which provides ratiometric calcium imaging, specifically expressed only in the maculodental cell in the kidney. And again, we do partial induction to look only at a single cell and apply intravital imaging over the time. I'm using, although calcium as an example, but obviously there are many, many tools that have been developed and widely used, including at Janelia. Other signaling cascades, cyclic KMP, Wnt beta-catenin signaling, so forth, and a variety of chemo and optogenetic tools. In our own team, we are using the DREAD constructs that provide excellent examples. So just to provide, again, intravital imaging examples for the endogenous activity of single cell types, using calcium as a readout, as a calcium for cell activity. I'll show you here to contrast the general bulk cell labeling approach and the single cell approach. On the left-hand side, I show you 2 time-lapse movies that show calcium activity in the totally intact in vivo kidney surface zoomed in on a single filter, the single glomerulus, using a general ubiquitously expressed Cree model, SOX2 driven Cree model that labels every single renal cell types, including the maculodental cell. So every single cell type has the GCAMP6 and SALSA to show activity. And if you watch this over time, this is about 10 minutes time elapse, fast forward, you see that there's one cell type which shows the highest level of calcium activity, which is our favorite cell type of the macula. And so here at the vascular pool. However, if you look at the recording here at the bottom of the screen, if you put a region of interest through the active zone of these cells, on many cells, you see the cumulative calcium changes show oscillations, overall oscillations over time, which we believe is important for the to regulating and directing vesicular exocytosis of important chemical messengers, for example, adenosine and ATP that act on contractile cells on the afferent arteriole to regulate blood flow. But you cannot see a single cell resolution, truly. So a horizontal view of the same cell plaque of the macula as is shown in the center image. Here you see the blood flow through the afferent incoming blood vessel and the efferent outgoing glomerular arteriole, and the cell plaque, the DMD cell plaque is right in the middle. You can clearly see individual cells and nice calcium transients in the single cells, apparently in a propagating fashion. But you cannot see detail. On the right-hand side, you can now have that this single cell induction. Now you can see this single cells of the macula here in contrast to the other approaches. And importantly, the recordings, now you can relieve the single nicely oscillating pacemaker-like oscillations in single cell. Same is applied in the confetti model. I know I'm over time, but to show that we need to have single cell labeling, tagging, to track the same cell type, fate, and function over time. We use the confetti model to label a variety of cell types, and again, label and track the same cell type, same tissue volume over time. Baseline, day 7 and day 13, in response to physiological stimulation. And I emphasize again the role of physiological stimulations, in this case, the loss of body fluid and salt, which helps tissue remodeling in this case. And we can observe clonal formations of cell types only in response to the physiological stimulation. So just to summarize, we can obtain key new tissue cell organ structure function information derived from this application of the single cell in vivo approach, which reveals new mechanism of tissue remodeling, organ to organ crosstalk. And obviously we can combine this approach with scRNA-seq for genomic approaches. Still, physiology is the key. And finally, I have only one item on the wish list, the need for deep tissue imaging to combine this with really deep cellular ability to visualize deep cell types. And we are very fortunate to collaborate with Chris Hsu at Cornell and apply 3-photon excitation, the use of 17 and 1800 nanometer lasers, which amplifies the tissue penetrating capability of about 3-fold by using 3PM. And so altogether, we need to develop and deliver single cell specific barcodes for cell ID. And we can combine this with other approaches, for example, single cell RNA-seq, perform for this single cell physiology over time and by the application of organ specific stimuli. Thank you so much for the attention.
Thank you so much, Janos. We'll now move to Catherine Schrenkel. If you could share your screen. Yeah.
Okay. Can everyone see this all right?
Yes.
Okay, and can you see my arrow when I'm moving it?
Yes.
Okay, perfect. Thank you. All right, well, thank you so much for the opportunity to present and I guess close out the meeting here to tell you about my work as a postdoc in the Hamdoun lab at SIO and UCSD about imaging the ontogeny and activity of small molecule transporters in gut epithelia. And I'll be talking about our model system, the sea urchin embryo and larva, obviously not drawn to scale here. But I'd like to begin by highlighting that depending on your team, I suppose, you have different answers to the question of what makes a gut. So here on the left side, we have physiology. So what makes a gut? You have very specific apical or basolateral membrane proteins that dictate the flow of small molecules in and out of differentiated tissue. This is done by small molecule transporters or SMTs. And also you have the capability to renew these types of systems. And here I'm showing you for gut epithelia in particular. Whereas here on the right for team developmental biology, what makes a gut is really the process. So you specify a field of endoderm, you make a tube, you pattern the tube, you make foregut, midgut, hindgut, so future esophagus, stomach, and intestinal regions. And depending on the time scale, you polarize and mature the tube. And this can happen at lots of levels, particularly with environmental inputs. For example, colonization of gut epithelia that dictates terminal differentiation as well. And so we've seen some beautiful talks throughout this whole meeting about both sides of the story. And I'd like to continue to argue that here at Janelia, you could really bridge these together with kind of a holy trinity of developmental cell biology, physiology, and imaging. And what I'd like to focus on is tracking and targeting SMT activity in vivo during gut development, function, and renewal. And I'm doing this with the sea urchin embryo, and so this is a blastula here. And before I introduce you to this, I just want to highlight some of the gaps in knowledge that we have on a very specific subset of these SMTs. And those are ABC transporters, or also known as multiple drug resistance proteins. And these are very important in having the cell deal with xenobiotic or foreign molecules. And in fact, they're often very highly upregulated in tumors to pump out Gleevec, for example, or other chemotherapy drugs. And despite their importance in terminally differentiated cell types, as well as in a tumor setting, we really don't know when and where they become expressed and active during the creation of the gut. And so how does that come to life? How do they get there? This can be at the genetic regulation level, trafficking to the membrane. And it's important to understand these because then we can understand how some of these early systems are recapitulated in gut renewal or even in disease states. And lastly, Elaine Fuchs yesterday brilliantly highlighted the problem of just the environmental challenges that we and really every organism are facing in terms of increase in pollution, changes in microbial environment. And so really, how do we test these environmental inputs and these early life exposures of these and how it impacts the development and maintenance of these systems? And so I'm starting to address these questions with the echinoderm system Again, these are sea urchin embryos, and I'd like to highlight some of the advantages. So first and foremost, we have highly synchronous gut development on the order of 2 to 3 days. So you can spawn millions of eggs at a time. These are fertilized externally. Endoderm is specified by 24 hours in the blastula stage. A rudimentary gut tube forms. We have very conserved gene regulatory networks. So all of these compartments are presaged already at the molecular level to become the esophagus, stomach, and intestines of the feeding larval. So these look like little spaceships, but what's wonderful about them is you can visualize the whole gut. We have a single epithelial layer. We have very specific digestive and pancreatic cell types that emerge post-feeding. And we can image these animals at a large whole animal scale. So we can micro-inject markers for your favorite markers, membrane, life act, tight junctions. So we can really visualize all stages of development. And finally, in the feeding larval stage, we can have very robust feeding and exposure assays. And so this video that I'm showing you here, we can see a feeding larva. This video is going to just move in Z space in and out of the gut. So you can see lots of this, for example, are immune cells that are migrating to the gut. You can see feeding. All of this structure is highly innervated. So there's a lot of systems that we can tap into. So one of the first things I did as a postdoc was answer the basic question, when and where are ABC transporters expressed? And in fact, this hasn't really been addressed in any animal model. And it was quite remarkable to us to find that some are very broadly expressed, such as the C subfamily and this ABCB1 protein, sorry, this is RNA. And others are very specifically expressed, for example, in the mesoderm and in the coelomic pouches, which is the future germ cell niche of this stage. And what I'd really like to draw your attention to is the transporters that are relevant in the endodermal tissue. And so these are genes that become enriched in the gut tube or are initiated very specifically only in specific compartments. So the next question is, okay, so we have RNA that's patterned. Where are proteins active? And I want to focus just on the B subfamily. So ABCB1 and B4 are homologs of human ABCB. And so to do that, we use fluorescent substrates to quantify efflux activity. And the way this works, it's actually rather counterintuitive, where a bright signal means low transporter activity. And that's because these substrates are often hydrophobic and they go through the cell membrane. And if there's low transporter activity, they'll collect in the cytosol. If there's high transporter activity, they'll be pumped right back out. And so we can use the variety of these substrates they don't have a single gene, single substrate level of specificity, but we can address subfamilies. For example, the B, C, and G subfamilies. And we also have inhibitors that are specific to these different subfamilies. And so when I do that, we see a lot of activity. So first of all, at the blastula stage, I'm showing you accumulation of this B-specific substrate, Bodipi-verapamil, and on the bottom panels are the membrane overlay. Again, these are confocal live, these are live animals. With the addition of the inhibitor of ABC-specific proteins, we can see that accumulation of the signal increases across the whole animal. This makes sense that they have high transporter activity. These are developing and floating in the ocean, pretty much encountering every small molecule and xenobiotic you could think of. And what's more interesting for the gut development is that at a stage where the morphology is quite evident, so here I'm showing you esophagus and midgut, there's very high activity already in the gut that's sensitive to inhibition, but these animals haven't even started feeding yet or using their gut yet. Once they do start feeding, again, you can see that different compartments have very high efflux activity. I've outlined where the gut compartments are. You can also see with the histone overlay. And again, this is sensitive to inhibition. And importantly, we can quantify this in different tissues by looking at the pixel intensities in different regions in single confocal slices. So we can also use these assays to validate our CRISPR-Cas9 mutants. And so when we use CRISPR to target the nucleotide binding domain, which is the region that is responsible for transporter activity, we can see reaccumulation of the signal. And again, this is quantified here. Now, what do these proteins do for the sea urchin in the gut? I was trained as an immunologist in my PhD, and so I'm very excited to learn about the crosstalk between microbial products. And so I can infect these animals with an opportunistic pathogen and then score the midgut pathology and infiltration of immune cells. And when I do that right off the bat, it's evident even before exposure that these animals have slightly thickened gut epithelia. And once they're exposed to these bacteria, both wild type and Crispins have gut pathologies and increased immune infiltrates, but these Crispins have it much worse. And they also have elevated conserved epithelial IL-17 responses. So this is really telling us that these ABCs are involved in the management of microbial-derived small molecules. And I'd like to highlight that the first IBD mouse model was in fact an ABCB1 knockout. But now, even 23 years later, we don't know what common types of substrates exist or mechanisms that are at play in transporter-mediated host microbe interactions. So I think this is a really wonderful space to explore, particularly in this model. And so just to end, I'd like to revisit this goal of having a systems level imaging of epithelial ABCs. And what I've shown you that we have in the sea urchin is fast development of the gut, an amazing transporter toolkit. We can now overlay this with gene regulation information. We will have transgenic lines, as well as encodable ABC-specific nanobodies. But what we need, in addition to that, is ability to multiplex more than one ABC at a time, both to localize subcellular trafficking and their activity. Again, more specific substrates and sensors, particularly in the bacterial world. And these all apply to a broader community. You know, your favorite organ has a lot of xenobiotic transporters that are important. And from an even bigger kind of 10,000 foot view, if anyone remembers planes, this feeds into remote sensing and signaling, so cross-organ movement of information, both at the adult and, I would argue, developmental side. And so I know I'm getting low on time, and just the last slide here is just very specifically where Janelia can help for ABC transport proteins. First, at the substrate level, substrates that light up would be really helpful with a physical interaction or concentration dependent. Again, a huge theme of this workshop is a small molecule world after all. Tracing transporter protein and activity in vivo. Again, there's over 40 transporter proteins, depending on your species. Can we multiplex the localization? Can this be coupled with trafficking machinery? We can also maybe have better ways to visualize activity from a cytosol perspective. And one thing to note is that this needs to be amplifiable, because oftentimes these are low copy number. You don't necessarily need a lot of transporters in the membrane to be effective. So we do have some signal to noise issues to work out. And finally, targeting transporters in a very specific manner. And the last theme that I think is very evident from this whole meeting is nanobodies are going to help this and all platforms we've been talking about. So here's my Rudolph, I guess the green-nosed llama giving us nanobodies. So with that, thank you so much for your attention. Sorry I went a little over time. I'd really like to thank my training environments, past and present, funding, and again, thank you so much for the opportunity to talk. And I'll leave you with this amazing comic. Nothing develops, responds, happens in a vacuum, and what a place at Janelia to really understand that at a granular level. Thank you, and we'll take your questions and discussion.
Yes, thank you so much, Catherine. Yeah, so we'll now move to the discussion Q&A session led by Sylvie Breton and Prabh Sengupta.
So while Prabh gather your questions and collects them, I'd first like to thank the speakers. You had a difficult mandate to surprise us after 2 days of amazing talks, and I think you succeeded in bringing new things. And I think that it was a perfect segue for the talks we've had earlier about kidney by Aura Weisz and Michael, and also some intestine here with Kat. So I would have a few questions, but I'll let Prabs maybe tell us what he's reading from the audience first, maybe.
Yeah, I think right now we don't have many questions, but there's a question from Daniel about like how the isotonic to hypertonic gradient in the kidney shapes the cell fate, morphology, and function. And I think Dennis answered it, but then Daniel followed up with asking whether transcriptional activator YAP is essential for cell fate determination in these cases. So maybe Dennis and Janos can comment on that.
Well, there's a ton of evidence from in vitro experiments, obviously, that hypertonicity changes gene expression and cell profile in the dish. But in vivo, it's a little bit less easy to do. There are changes in cellular profile. If you the kidney, of course, throughout the day, the cells in the kidney are exposed to different levels of tonicity in the medulla. Sometimes it's more, sometimes it's less, depending on your hydration state, and also depending on medications. So, for example, somebody taking diuretics may have a medullary osmolality that's as little as 300 to 400, whereas somebody who isn't may go up to 1,000 milliosmoles per kilogram. The major thing that we notice in the kidney collecting duct when the hypertonicity, if you like, it goes away, is that the cell profile of the collecting duct changes a little bit. There are more intercalated cells, the acid secreting cells, and fewer principal cells in the collecting duct. And in things like animals with nephrogenic diabetes insipidus, in other words, with who produce a lot of urine, they wash out their gradient. They have many more intercalated cells than normal animals do. But that's about it. I don't really know of any major structural consequences of this. The animals live okay, they just drink a lot. And so there is a transcriptional change because the cells exposed to hypertonicity start expressing genes for organic osmolytes like sorbitol and taurine and things like that in order to balance the tonicity in the interstitium. But other than that, there's really not a big detrimental effect because the kidney's used to it.
Okay, so looks like right now we don't have any questions. So maybe I can start off with a question for Janos, and maybe Dennis can also pitch in. Like, Janos, in your talk, like the concept of chief cells driving the response of organs, tissues to specific stimulus, that's very intriguing. So I was thinking, how universal do you think this is? Like, would like other organs like liver or intestine have similar kind of cells that act as like a central decision maker? And then maybe the identification of such cells in these different tissues and enumerating how they function, that can be in principle a very exciting research avenue, right?
Yeah, so this is a great question. And I think this is totally applicable, and there's literature for this, totally applicable to other organs. So some of this was actually unpublished that I show you today, but the concept is clear and has been confirmed by many organ systems. For example, the liver has been published nicely, and other GI organs, that fasting, intermediate fasting, and referred today before, is really acting on some precursor cells or even progenitor cells, stem cells to mediate tissue remodeling. And the importance, the clues are the drivers. So what are the organizers, cell types, or the drivers of this stem cell mediated remodeling in the specific tissue environment is the question. In our case, we believe that this important mysterious cell type of the maculadenza in the kidney is playing an important role. There seems to be analogous cells in other organs. One important point is that I showed you some calcium imaging as one of the many potential functional readouts of these cells. Mm-hmm. And part of this, I'd like to link to another talk we heard yesterday about TMEM16A, and that some other cell types throughout the body express this important voltage-gated chloride channel, which has an important role in mediating calcium. So the same molecule, an important building block of all types of pacemaker cells throughout the many different cell types in many organs this is an important example. So it is there in the maculadenza cell, again, providing this pacemaker-like calcium activity that provides a propagating calcium signaling from cell to cell in a lateral dimension along the monolith epithelial layer. The similar is there in the gut, in the gut local nervous system in the gut, providing for nice GI motility. The same could be playing an important role in the airway. We've heard beautiful talks yesterday about the mucin secretion and the laminar flow of mucin from in the airways. The similar pacemaker cell, calcium pacemaker mediated pathways may be important in role, and the same cells may be important in tissue remodeling on a more chronic fashion as another layer of function.
Thanks, Yanis. We have a question from Alan K. in the question answer. It's for Dennis and Yanis. He's asking, do you think it might be possible to dissociate complete intact nephrons?
Yes, it is. Well, first of all, it's not easy, but it's been done many times, dissociating intact tubules. So tubules can be isolated, and people have micro dissected intact nephrons, and even gone as there are even proteomic profiles and gene profiles of nephrons that have been isolated and chopped up into segments. So yes, it is possible. Yeah.
Okay, I see Michael has his hand raised, so maybe we can bring him in.
So all three talks were wonderful. My question is for Catherine. You sort of prefaced your talk by indicating that these ABC transporters might be involved, at least in part, in responding to environmental toxins or protecting the organism against them. I'm wondering, have you ever looked to see whether they are induced by exposure to environmental toxins? We know, for example, that the first ABC transporter that was really identified, multi-drug resistance or P-glycoprotein, is dramatically induced by exposure to chemotherapeutics. Mm-hmm. Is it possible to understand what a sea urchin likes and doesn't like by seeing what ABC transporters are induced in response to changes in environment?
Yes, so that's a great question. Thank you. So the short answer is yes, they definitely have the capability to respond. And I think that is what makes the system so wonderful, because all of the environmental response of these proteins have really only been studied in terminally differentiated epithelia or in the tumor environment, as you said. And so I do think those areas are open and ready to be, reactive, or even in the developmental stage. So, for example, there's now single cell sequencing of early development up until the formation of a gut tube. And we're actually mapping where the nuclear hormone receptors and other kind of inducible receptor systems that would be our candidate regulators of these proteins. And so especially for animals like the urchin, I think they definitely sense their environment while they're developing. And I'm doing experiments now to test how much they change the either expression patterns or intensity of expression during development before they even feed. So yes, it's a really exciting area that we're following.
Thank you.
Catherine, so Catherine, I had a related question to Michael. I was thinking, like, this patterning of these ABC transporters, do we know anything about if they are patterned in like epithelial layer of the airways of lungs? And is there any kind of change in expression pattern to environmental exposure, like if somebody smokes or you have asthma?
Okay, so you've stumped me. I don't actually know in lung epithelia, but in the blood-brain barrier, definitely. In the kidney and other epithelia, yes. And for the blood-brain barrier, there's actually work that has shown glucocorticoid exposure from the mother actually will change the output of expression in the fetal of these transporters in the fetal blood-brain barrier. So there's a lot of crosstalk between environmental inputs changing the expression of these transporters before they're even kind of used to protect from other types of environmental xenobiotics.
So on that topic, Catherine, I think maybe I missed it, but I think you're one of the few speakers who actually talk about immune cell recruitment towards an organ. So, and that's one reason I think that it's essential to study epithelial cells while they reside in their native environment. So there's so much crosstalk between the epithelial barrier and the immune system. So for one question, you mentioned the ABC transporters are there to sense microbial derived substances. So what would be these substances, and how do immune cells know they need to be recruited into the intestine in your system?
So I'll kind of answer that backwards. So first of all, how do immune cells know where to go? Yeah. So what happens, so the pathogen that I was using is a Vibrio that collects in the gut. And what happens is the gut becomes inflamed and it initiates an IL-17 specific epithelial response just in the gut epithelium. And that leads to downstream signaling and, you know, kind of like a black box question mark arrow in terms of the signals that then are released and recruit the different immune cells to infiltrate and get to the gut. So when you knock out the receptor, IL-17 receptor, for example, you don't get the right type of signaling. So it's definitely something in the gut is sensing that there's a problem, and then it relays that to the rest of the animal, like help. And then in terms of what are the microbial metabolites? So the short answer is we don't know. There's a lot of, I want to say, phenomenological evidence that these transporters transport either toxins or metabolites, kind of secondary metabolites that are made. A lot of, so for example, in certain other enteric diseases, I'm blanking on the exact bacteria, but they will actually downregulate the host transporter. So there's definitely, the host transporter is doing something that the bacteria doesn't like. So that's something I want to identify and see, are there common principles of bacterial molecules? Probably things that are hydrophobic, things that are, there are some biochemical things that would be unique. So the short answer, don't know yet. We're working on it, and that's a big, I mean, it's relevant for our animal, for humans. So we're excited to understand that crosstalk.
I have a few ideas. We can talk offline. I'd like to give my colleagues time to talk, but we can talk about that if we run out of time today. Otherwise, I'll come back to that.
So we can probably go to an interesting question from Aura and Manu. So they are asking Janos and Dennis, like, do you think studies in extreme animals might inform our overall understanding of kidney function? Example, in snakes that eat only once a month, or camels? And what would the evolutionary biology approaches or comparative physiology studies be more informative for understanding these things?
I think we can both have a go. Janos, you want to go first?
That's an excellent question. I'd like to, I can give my few cents first, but excellent question. So I would love to put a snake on the intravital microscope and other extreme organs. Being in the Southern Californian area, I even had the plan to go out on a road trip with my lab and collect desert shrews which have very long medulla, extreme fluid conserving capacity. So there are wonderful examples out there, and evolution is exactly very relevant, totally relevant to physiological control, physiological adaptive mechanisms needed control of tissue remodeling and obviously many other organ functions. So in fact, in translating this exactly, for example, to the capacity to conserve body fluid and salt as a major driver of the complexity of the nephron. As we know, it developed to the mammalian system from a very primitive fish system to reabsorb and conserve more body fluid and salt with the evolution. The detachment from the sea, salt-rich seawater life to the dry land and salt-less conditions was exactly the driver of these morphological and functional adaptations that drove tissue remodeling, if you wish, for the complexity that, and it's exactly the same physiological adaptive pathways are there to remodel tissue regeneration, if you wish, even in all of us right now. All these mechanisms are actively at work. So that's where this specific cell type that I was talking about as the major salt sensor in the kidney, the macular cell has a major contribution. So extremely relevant. So thank you, excellent suggestion.
Yeah, I think a lot of the early work on vasopressin action was done actually at the NIH and other places on the toad urinary bladder. So toads, toads go into water, they soak up water through the skin, and then they hop onto a rock and they wait for a dragonfly to come along to eat. The sun's beating down on them, and they have their own Nalgene bottle. It's called their bladder. And so when they dehydrate, they express, it's called vasotocin. It acts on the bladder wall, and it causes the fluid to be pulled back into the frog. And so they have a very, very similar water channel response to the collecting duct. So the toad bladder is really a collecting duct, and it also has the proton secreting cells as well. So a lot of the early work was done on that modeling system. We in our lab, Janos, we actually did go out, Alfred Van Hook in the lab went out and got licenses. You have to get a license to do this. And he caught Samomys obesus, the desert rat, or the kangaroo rats in the desert. You can only catch a certain number a year. We did it in conjunction with some people in Arizona, because, as you said, they've got this giant concentrating mechanism. They don't drink. They live entirely on plant and on seeds. And if you put them in the lab, we got them in the lab, they just don't drink. You can feed them on seeds, and they just extract all the moisture they need from seeds. And the ones that we had, if you actually give them something to drink, they go wild, and they kind of get edematous because they love drinking so much, but they don't do it normally. And so they have a problem excreting water, not maintaining it, but excreting it. When we looked at their kidneys, they have no aquaporin 4. And we don't know why. It's like an aquaporin 4 knockout. That's the only difference that we could tell. No aquaporin 4 whatsoever, and they concentrate like mad. So that's one thing that we did. The other interesting animal to look at is bears. Bears, when they hibernate, they get huge levels of urea and nitrogenous waste products in their blood, and yet it doesn't affect them. And if we had, if we had that level of nitrogen in our blood, we would need dialysis immediately. But they don't. So that's one thing that people are looking at. Let's see. Oh yeah. The other thing I'd just like to say at this point, in response to a question that I think Ron asked here, how long does an intact nephron survive in ex vivo, was it depends on the nephron. Collecting ducts are quite short-lived, just an hour or two. Sorry, not collecting ducts, proximal tubules, but the collecting duct can live several hours. And here's kind of something that in the old days of last century, lots of labs did tubule perfusion experiments. Now, because the NIH stopped funding that type of experiment, there are very few labs that actually do perfused tubules. And getting somebody to collaborate with you on tubule perfusion, you have to wait in line as if you're waiting for a COVID vaccine. It's very, very difficult to get a collaborator with tubule perfusion. So that's an old technique that's becoming very useful again now, but we don't do it enough.
Yeah, Nusse does it.
Yeah, I know. He's one of the few.
Yeah. So we have 3 people patiently waiting with hands raised. So maybe we can go to Jens first, and then Michael and Manu.
Yeah, thanks. Catherine, I'm a big fan of all model organisms, but I didn't realize that yours in particular, I guess they all do, underwent this transition of sort of pre-feeding to preparing to feed to feeding, which is essentially what placentals do in utero. They can swallow as fetuses, but they're obviously not having nutritional, it's amniotic fluid. So I wonder if you have compared transporter changes more broadly in that transition of pre-feeding to feeding with, say, a placental or something, that undergoes the same transition. And reason being is that preparing the gut for food before birth is essential. And if that's not done at the right time, usually it can lead to a lot of really debilitating downstream consequences. So I wondered if you can actually use this system to study that transition. And just as an aside, I don't know if you mentioned, but do they have a microbiome? I may have missed it. I'm sorry, I had to step out because my transporters were overactive, but either question's fine.
Right. I'll go backwards again. So first question, do they have a microbiome? Yes, they really are only colonized in the gut once they begin feeding. So there is opportunity to kind of really look at transporter function pre and post, both feeding and commensal. It's a lot of proteogamma bacteria. And yeah, there's a lot of work in the literature looking at the specific microbiome types. And you can kind of make azenic ones as well. So germ-free urchins, which are very interesting. They actually grow much faster when they're totally germ-free. But then if you infect them, they can't deal with it because they're same with germ-free mice, just their immune system hasn't been educated properly. So we can do a lot of parallel experiments. As far as the placental stuff, I don't think so directly with the urchin, but there are people that are looking at transporters in the placental barrier. For example, Saloran Alexunis does that. And I'll add something to the chat directly, one of her references. But in terms of fetal gut ABC activity, it's just, we don't know yet because it's so hard to study, certainly in humans or even mice. But that's another reason why this environment interaction is so important to understand, because there are, for example, polybrominated compounds that are from flame retardants. These are very high level pollutants that if you took your own blood, you would find in your own blood system. It's in cord blood. We know the fetus gets exposed to them, right? And they can physically, what Amro's lab has shown is those can physically bind to transporter proteins, so the ABC transporter protein. And when it does that, the transporter can't efflux other toxins as well. And so understanding the most vulnerable windows of fetal development, neonatal development, postnatal development to those types of compounds is something we're thinking a lot about. And hopefully that in part answered your question.
Manu, probably you can go next.
So I just want to make two brief comments. One is we go out and we collect with plankton toes lots of starfish larvae that we've been studying for a while. And over the years, we find, and again, I think, based on the kinds of experiments of exposure to microplastics and the context of, I just want to highlight this idea that cell biology at the interface of environmental degradation, we know so little about the kinds of environmental perturbations that are happening and evolutionary processes that are happening for the very first time, because many of these materials might never have been exposed. So I'm just curious, this is a broader picture question of what we call normal physiology is changing dramatically in our ecology. What we call normal ecology is dramatically changing. And then the other thing that I resonate a lot with, but I want to also challenge the community, it is extremely difficult to work on, this is why marine stations work. You can go get a license. We have a license for plankton tow. We can collect many things as long as they don't have spine, and study and understand and do comparative biology. But the idea that Dennis described and many of the terrestrial systems that are equally remarkable, it's extremely complicated to put together a kind of an infrastructure for exploration, because you don't know what you will find. And, this is exactly where places like Janelia could be the terrestrial zoos, but from a cell biological perspective. A lot of physiology, zoology departments, you can get specimens. We go to museums quite often. We try to actually go to autopsy reports to get some tissue samples. So I'm just curious how people think about it as a community. A resource like that would be remarkable. It is non-trivial, but can we have kinds of commitments that allow us to this was the heart of physiology. Can we return to that?
Of course, my answer is yes, particularly in marine invertebrates. And, everything we know about neuron transmittive signals is from sea hares, because they have the giant neuron. So I think, maybe if there were directives from NIH or NSF even to establish a long-term commitment for funding these marine stations, so SIO, Woods Hole, Friday Harbor, especially when we have such tumultuous federal funding climates with the recent election cycles.
Much of what we know about cystic fibrosis transporters was done in the shark rectal gland.
Yeah, I know.
Right.
Yeah, so long-term funding for those types of stations, yeah.
I guess the incentive structure for research funding has to also change, right? For this to be successful. That's one of the problems we face, right? So maybe Michael can come on in. I think he had his hand raised.
Yes, just, first of all, to follow up on what Manu said, we are the proud owners in our department of what I believe is the largest hibernaculum either in the country or on the East Coast, because Elena Gracheva hibernates ground squirrels. So I'm a very big believer in the idea that comparative physiology is illustrative of physiology in general. And I think the idea of identifying and keeping species that can be informative is wonderful. My question was for Janos and Dennis, and it's sort of a Janelia-esque question. You know, neuroscientists have the wonderful advantage of being able to exploit optogenetics to turn things on in very specific locations. Janos, you alluded to using dreads, and I think that's a great idea. To some extent, in the kidney at least, and in most of the other epithelial organs we've talked about, optogenetics is not very useful because of the either physical scale or redundancy. In other words, if you want to turn something on very fast in a small space, it's going to have very little effect on physiology generally if you activate 5 nephrons in a kidney that has 1 million. So I'm wondering if you have thoughts about how to temporally control physiological properties in a way that can be informative as to how signaling networks affect global physiological processes.
Yes, Michael, this is excellent question and right on point. I think even before we go to optogenetics, intravital imaging is a great example for this approach, because we've been using the laser light as a micromanipulator even before optogenetics became available. So you can actually use the exciting laser to do pretty much anything, stimulation or actually even nanofabricating something in the tissue to manipulate the tissue, specific cell types, even subcellular organelles within a single cell. So that tool has been available, but now you can combine it with optogenetics. I think manipulating and perturbing some of the nephrons, single cell types again, perhaps a few nephrons, I think that's very consistent with the theme that we've been developing as a panel, that we need this to better understand how individual cells function. And that laser or optical optogenetic manipulation clearly has that advantage, and it's a great tool. And in our specific cell line of the maculodensa, which is now we understand this is actually a neuron-like cell line, I think we are in great shape to use all those tools that neuroscientists have developed, as you mentioned, and that's all immediately and readily available to us. So I think this is a great point and potential applications.
Yeah, I think Janos is right. I mean, the applicability of caged compounds and of photoactivation is probably going to replace the old studies with single nephron micropuncture. So this is the new single nephron studies, right?
So I think we have time for one. Oh yeah, Sylvie.
No, that's okay. I just I'm just looking at the time. Let's give Francis the chance to. Yeah. Her hand was raised.
Oh, me.
Yeah, you.
Yes. I just wanted to echo the idea of looking at the animal which best, as a good example for your system. So the use of different animals. But at the same time, if Janelia is going to do this, it might also be valuable for conservation work, because many of these animals which were used in the past are actually reducing in number. And one of the things I've been horrified by is what's been happening with the limulus king crabs, which are so much less these days, and especially which are being used, for their blood in a very industrial way. So maybe Janelia could also keep some track of that as well.
True.
Yeah. Well, that's a really great point to end our session on. Just, what are the different organisms that we need to keep our eyes on? And, yeah, it's pretty profound. So if I could get Michael and Patrick on, I want to thank the Probs and Sylvie, as well as Dennis and Janos and Catherine, for a terrific last session. Very, very exciting to hear about, all of the different single cell and renal physiology that we just heard. But over the next couple minutes, I thought Michael, Patrick, and I could sort of draw some concluding remarks, and I'll start and just say that it's really been amazing the last 2 days. Just incredibly stimulating ideas that have been pushed back and forth. I want to thank all the panelists, the discussion leaders, and the outside participants. It's really been a very stimulating experience. As you all know, we've covered a huge amount of material related to epithelial physiology and its underlying cell biological basis. And what's clear to me is that any understanding of 4D cellular physiology is going to have to consider the many roles of epithelia in their forming the boundaries of organs, in their allowing organs to sense and integrate chemical and mechanical cues, as we heard, in their patterning the structure of an organ, and finally, in their ability to coordinate how organs function in health and disease. So with all the new insights, ideas, and approaches that we've heard about the last 2 days, it's clear that efforts at sharing and coordinating this understanding of epithelial physiology is going to have to be manifest. And so I really want to urge everyone to continue our discussions and interactions in this exciting field. I'm hoping that as Janelia moves into the 4D cellular physiology world, that we're going to be able to continue this kind of discussion, especially among people like yourselves who focus in the epithelial realm. So I now want to turn over to Michael and Patrick for their comments, but just thank you all so much for coming. Michael, why don't you could start?
Sure. So I want to echo everything that Jennifer said, to thank all of the participants, to thank the outside participants, to thank the AV people who accomplished the miracle of making a Zoom conference with this many participants actually vibrant and not annoying. And I also want to thank Janine, who did a remarkable job of pulling this thing together incredibly rapidly and going from concept to completed program with grace, speed, and calm, which I give her a huge amount of credit for. Yeah. I also want to echo Jennifer's point that epithelia are, pardon the pun, but the beating heart of physiology. I think that in order to understand physiologic processes, you have to understand the role of physiology. And I think for the purposes of making it communicable and palatable to Janelia, I think it's safe to say that neurons are really just funny-looking epithelial cells that have maybe taken their polarity to something of a length extreme, but axons and dendrites actually sort proteins through very similar mechanisms that epithelial cells use. Epithelial cells are transporting substances, neurons are transporting information. Epithelial cells serve sensory functions, can actually talk directly to neurons and receive cues from them. So I think that understanding the ways in which epithelial cells create what Claude Bernard called the milieu intérieur and protect it, and the ways in which they talk to and respond to the nervous system to maintain Walter Cannon's homeostasis is a tremendous goal for 4D cell biology. So Patrick, why don't you wrap it up?
Great, yes. Yeah, I just don't think enough thanks can be given to especially all the panelists for their participation. And we definitely asked for an out of the ordinary time and effort commitment. So it's just been really great that it turned out so well. Yeah, I think Jennifer and Michael really covered the importance of the epithelial really well. And I think another theme that emerged is sort of this tool development idea. And it seems that everyone really just wants a way to observe their biology that they're interested in. And the question is really how to enable, a broad usership for that. I think in an ideal world, that would be, creating a broad library of genetic mouse lines or genetic organism lines of your interest. But, I think if we just consider the entire budget of the 4DCP, we could probably make 25,000 mouse lines, which would about cover all of the genes. But unfortunately, we do have to pay postdocs and grad students. And so really, I think it's going to require some sort of compromise in which we can develop tools that allow us to observe and then also analyze our biological process of interest. And luckily, I think Janelia is really well positioned already with the microscopy advances and the computation aspects already. So I'm pretty excited. I think they can really make really have a toehold into this field. And then, yeah, I guess I'll just wrap that up by saying thank you again.
Yep. So this concludes this workshop. Again, I hope everybody we can continue discussions in this area because it's really amazing how the potentiality that we have, the synergy among all of the different people that we've just discussed that have just talked to us. So let's keep in touch. And for all the panelists, unfortunately, you're going to have to spend another hour with us. We're going to sort of brainstorm some of the concepts that we've heard. But for everyone else, thank you and have a great rest of your week. Bye-bye.